Good morning, all of you. I request Dean Sir, Dr. Virendra Minke, Sir, Vice Principal Ranjit Moite Sir, PhD Director Hausarao Patil Sir, and Hospital in charge Vishal Patil Sir to for Dhanvantari project. Good morning, all of you. Myself, Dr. Dipali Prasad Patil, Associate Professor, KRIYA SHARIR Department, welcome all honorable dignitaries, delegates, teachers, scholars, and all students on behalf of Loknete Rajaram Bapu Ayurvedic Medical College, Hospital, PG Institute and Research Center, Islampur. On the occasion of birthday of Honorable Nishikant Bhosle Patil Dada, founder Prakash Shikshan Mandal, Department of Kriya Shari is honored to arrange national webinar on genetics and Ayurveda. Wish you many happy returns of the day, Honorable Dadaji, on behalf of Loknete Rajaram Bapu Patil, Ayurved Medical College, Hospital, PG Institute and Research Center, Islampur. I request Dr. Musharraf Sayed Sir, Professor and HOD, Department of Kriya Shari, to give preface of today's webinar.
Good morning, all. On the wonderful occasion of birthday of Honorable Nishikant Dada Bhosle Patil, founder Prakash Shikshan Mandal Islampur, Department of Kriya Sharir, Loknete Rajaram Bapu Patil Ayurvedic Medical College, Hospital, PG Institute and Research Center, Islampur, is honored to arrange the national webinar on genetics and Ayurveda. On behalf of our institute, I heartily welcome today's eminent faculties, Dr. Sudhir Pawar sir, he is professor and HOD, Department of Anatomy in Dr. Vittal Rao Vikhe Patil Foundation's Medical College, Ahmednagar. Dr. Aniket Shilwan sir is Assistant Professor, Department of Kriya Shari, G.J. Patel Institute of Ayurvedic Studies and Research, Anand, Gujarat. I also welcome Dr. Anura Bale Madam. She is Principal, Professor and HOD, Department of Kriya Sharir, Gomantak Ayurvedic Mahavidyalaya and Research Center, Shiroda, Goa. I also welcome our Dean Sir, Dr. Virendra Minkine Sir, our Vice Principal Sir, Dr. Ranjit Moite Sir, our PG Director, Dr. Pramod Kanab Sir, our PhD Director, Dr. Hausarao Patil Sir, our Hospital In Charge, Dr. Vishal Patil Sir, and our Director, Prakash Shikshan Mandal, Mr. Sandeep Yadav Sir. I also welcome all the respected faculties and all PG and UG students joined over here. This webinar is arranged to explore the knowledge on an interesting topic, the genetics, with the both views, modern and Ayurveda. Bija, Bijabhaga, Avyava are the terms used by the ancient Acharyas while considering the genetic hereditary defects. Acharyas also described some of the diseases like Prameha with regards to defect in genetic components. Kulaj Vyadhis and even the Prakriti, the concept of Prakriti is also having the basis of genetics in Ayurveda. Today's two eminent speakers will guide us on first role of genetics in medical education by Dr. Sudhir Pawar sir, and secondly the Prakriti and genetics by Dr. Aniket Shilwan sir. Dr. Aruna Bale madam will give expert opinion and summarize both the sessions. So I hereby request our Dean Sir, Dr. Virendra Minkire Sir, to say a few words about the today's webinar. Thank you. So,
Good morning to all. I, Dr. Virendra Minkire, the Dean of L.R.P. Ayurvedic Medical College, Islampur, District Sangli, Maharashtra. Extend my warm greetings and welcome you all on behalf of our institute, management, staff, and students to this national webinar from the Department of KRIYA SHARIR on the most interesting topic, Ayurveda and genetics. I also welcome today's distinguished eminent speakers on this topic, Ayurveda and genetics, which helps all the fraternity of Ayurveda to understand the concepts of genetics explained in our Ayurvedic classics. I wish all the best to the staff of KRIYA department and to the speakers. Thank you, one and all.
Let's move on towards today's first session of webinar to be guided by Dr. Sudhir Pawar sir. Honorable Dr. Sudhir Pawar sir is working as Professor and HOD in Department of Anatomy of Dr. Vittalrao V.K. Patil Foundation's Medical College, Ahmednagar. He has completed his MBBS from BJ Medical College, Pune and MS Anatomy from Grant Medical College, Mumbai. He has huge teaching experience of 22 years. He has also adorned designations like Nodal Officer in CME, Nodal Officer for CET exams, Resource Person for CME, Life Member of Anatomical Society of India, Society of Clinical Anatomy, Maharashtra State Chapter of Anatomy, BOS member of PIMS Loni and DY Patil Vidyapit Pune, executive member of ASI. He has developed cranial nerve museum separately. He has published 22 research papers in various national and international journals. It will be privilege for us to invite Dr. Sudhir Pawar sir on this platform as a speaker for this session, Genetics Role in Medical Education.
Hello. Hello. Hello.
Hello, sir, you can share the slide now.
Uh, can you see, sir, my slides?
No, sir.
I have to go on the screen share, na?
Yes, sir.
Is it visible?
Yes, sir. Yes, sir. Now it is visible.
Okay.
Okay.
My all slides is seeing or one side it is blocking?
Yes, sir.
My slide is completely slide is seen.
Yes, 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 sir. Complete slide is visible.
Okay, one.
Yes, sir. Now slides are visible.
No, no,
One by one.
Hello.
Slides are visible, sir, one by one.
But I'm not able to see from my side because some videos are blocking. 1 minute. I have to close it. Hello.
Yes, sir.
1 minute. I will mute myself. I will clarify within 2 minutes because videos are blocking my slides from my side.
Okay, sir. I will call you.
1 minute. Hello.
Yes, sir.
आता दिसते सर स्क्रीन.
सर हो स्क्रीन आणि साइड व्ह्यू पण दिसतोय.
1 मिनिट. आणि व्हिडिओ दिसतोय का माझा? नाही ना?
नाही व्हिडिओ नाही दिसत सर.
1 मिनिट. व्हिडिओ मी चालू ठेवायचा का बंद ठेवायचा?
व्हिडिओ तुमचा
हा.
चालू ठेवला तरी चालेल, काय अडचण नाही सर.
मैं बंद करता हूँ आपको नहीं दिखना चाहिए हाँ बाकी का नहीं मेरा इधर कॉम ठीक है टाइम जा रहा है हॅलो
येस सर.
हाँ दिसतोय माझा स्क्रीन विजिबल आहे ना
हां सर.
okay so I will start now
ओके ओके सर.
good morning everyone myself I am Dr. Pawar I am working as a professor head here in the Department of Anatomy in the Dr. Vithala Vike Patil Foundation's Medical College Viradghat Ahmednagar. As far as today's the national webinar on the genetic is considered, I am very thankful to the Dean sir of this Ayurvedic college, then organizing secretary from the SHARIR KRIYA, means the physiology department, and all the respected dignitaries. As today's topic, what I was asked to overview of the role of this genetic in the medical education. So this genetic is a branch of the medical science, and it is a very big branch, right? As nowadays, the importance of this genetics we are coming to know. So I just will brief on this, what is the role, right, of this genetic in the present era in this medical education. Now, when we are briefing on this topic, so I would like to go on this, the objectives, what we can say, right? So what is the genetic, then area concern in the genetics, what are the various corners they are expected in this genetic, then genetic in the medical education. So how it is genetic for the undergraduates, for the postgraduates, and overall, when these students, they went in the society, right, and they're doing practice. So what is the situation of the genetic at that level? Then we'll see at the end, or we'll emphasize what is the role of this genetic in medical education. And at the end, we'll see genetics, what is situation in the present and what will be the situation in the future medical education. Coming to the first point, what is a genetic? Now, the genetic can, as already I explained you, genetic is a big branch. It is a branch of the science, and this deals with mostly the genes. So one can define this, the genetic, it is the study of the genes and of statistical laws. There are certain laws in this genetic which are concerned with the inheritance that governs the passage of genes from one generation to the next. Now in social life, routinely, that when we observe around us, usually when we see the small child, right, usually we see the characteristics and we say that this child is right, mostly looking like mother or mostly looking like father, or these are the characters, right? They are similar like mother or father. What does it mean? If you go in the details of this, they are looking like right, mother or father, means there are certain characteristics. It may be physical or it may be developmental, right, psychic. So they are comparing, or they are more or less similar like mother and father. So here is the root of the genetic. So they are looking like means they're coming from parents to the offspring. So in definition, we can see, right, and this is genetic. So the study of the genes and of statistical laws that governs the passage of genes from one generation to the next generation, means parents to the children or offsprings. Now in this, the important terminology one should know, inheritance or heredity. Means what are the characteristics they're passing from one generation to the next generation. So this is overall in genetics we call as inheritance or heredity. So it is nothing but the process of transmission of characters from one generation to the next generation, or the characters from parents to the children in simple words. So overall this governs. So this will explain you, right, what does it mean by the genetic. Now there are certain branches, right, of the genetics, because genetic is a very big itself science, and there are sub classifications. We can see in the genetics there are sub branches. So in that again they are working on the specific points, right, in the genetic, and they are like this. So sub branches of genetics that we'll get, first is the cytogenetics, where the study of chromosomes is done in detail. So that branch particularly, or we can say the sub branch of the genetic, it is labeled as cytogenetics. Then comes the molecular genetic, again going in detail, right, depth of this chromosomes. So study of chemical structure of gene is done at the molecular level. So this separate branch, right, sub branch of genetic, we can say, right, or it is labeled as a molecular genetic. Then the developmental genetics, where the study of genetic control of embryonic development studied. Now, once the fertilization occurs, the zygote get formed, and this zygote, there are multiple cell divisions are going on, and they will form, right, the from single cell to double cell, and then, right, the multiple cells, millions of cells. The cells comes together, they form tissues, then tissues forms organs and system, and the particular species get formed. So all this development, so at the end of study, right, it comes to know it has all control or governed by the genes. And this is separate study, right? So that is embryonic development. And this is sub-branch labeled as developmental genetics. Means where the study of genetic control of embryonic development is done. So the fourth one, the sub-branch that concerned with the genetic is immunogenetics. So where the genetic control of production of different antibodies. Now, immunity is mostly, right, in case of immunity, antibodies are formed in our body whenever there is infection, right? So these antibodies, also the production of it, what antibody should produce, where it should produce, what is the quantity of these antibodies, and how it should be produced, the manner. So all this is controlled. Again, there is a gene control over that, and this specific or the separate sub-branch, right, labeled as immunogenetics. Then there is a fifth one, right? The subdivision or sub-branch of the genetic, we can say it's a behavior genetics. So influence of genes on the behavior of an individual. So psychological, what influence of that particular gene is transmitting from parents to the offspring or the next generation is studied separately. And this is labeled as the behavior genetics. The sixth is the population genetics. So overall, the study of this, the frequency of genes, right, in certain population is done, and what is the rate of mutation in such population it is studied. So this branch, right, is separately in the genetic level as population genetics. Then the seventh one, sub branch, we can say, right, it is a biochemical genetics. So where the study of enzymes, right, so how they're produced, what is the gene, what are the genes concerned with that enzymes. Basically enzymes that are prepared, they formed by the protein, and these proteins synthesis is under control of the gene, and these enzymes are needed for the various metabolism in our body. If these enzymes are not produced, right, we get the various errors in that particular developing baby, and which are labeled as inborn errors of the metabolism. So basically this is concerned with the enzyme study, and these enzymes production is under the control of the gene. So overall, if you see all these, right, all the 7 branches, so the cause or the root is going toward the genetic. So genetic has a very expansive role as far as the medical fraternity is considered. Previously, it was neglected, or most of time, right, it was not known. Research were very less in that. But nowadays, as the advanced research is going on, so we come to know, right, how the very expansive role of the genetic in the present. Take any diseases, right, cause is going toward the genetic, cause is going toward the gene, particular gene level, right, all these, the causes of the various disorders, they are there. So in this way, right, so these sub branches, one can, right, say that, like cytogenetics, molecular genetics, developmental genetics, immunogenetics, behavior genetics, population genetics, and biochemical genetics. So they are the various branches. So first we have seen the introduction, definition of this, the genetics. Then we have seen inheritance, right, or the heredity means what, and then this various sub branches. Again, we can emphasize what is the importance of this genetic, right, in this, the medical fraternity. Our present knowledge of genetic has its root in Mendelian principles. Yes, Mendelian, right, when someone discuss, right, with the genetic, we cannot forget the name of this, the Mendelian, right, who is known as the father of this genetics, right. So his initial name is like a John Mendelian. Later on, right, he adopted his name as a Gregor Mendel. He born in the July, right, 1822, and died in 1884. So he had a very important, what we can say, right, milestone, right, developed on the genetics. When he worked as a priest in some church, right, so he had done a lot of experiments, right, on the pea plants. He observed, he had done a lot of observations over this, how these pea plants are growing, then tall size, small size pea plants. You observe, and afterward, right, you work out over that, right? Few plants are there tall, few are small, and if you done right some crossing, so what is the next generation? So you observe for many years, near 6 to 7 years, right? Work out over that, and then he comes to the certain laws. So there's a these are called as statistical laws, right, of the inheritance, and it has a very important value in this genetic. So overall, his experimental study on the pea plant's growth, right, he stated laws, and they are known as Mendelian's laws. He stated it, right. Later on, most of the scientists come to know, right, so what the work he had done, right, over the genetic, and how it is very important as far as considered inheritance or heredity. Before this all experiments, the importance of nucleus in cell division was not clear. Chromosomes were not discovered. Details about the cell division not known. It was also not known that hereditary factors, so overall we call hereditary factors, and they are concerned with the genes. They are present in chromosomes. So it was all the mystery, right? So mysterious things, right? So nobody knows. They don't have the clear idea. But he devoted his life to work over that, and he had given, right, some clues, some direction, right, how this genetic is working, and what is the role of that particular hereditary factors or genes, right, which are present in chromosomes, right. So it is very important discovery as far as consider this the Mendelian's laws, right. In recent time, our attention has shifted from infectious diseases to the genetic disorders. Nowadays, in present era, right, so there are so many infectious diseases, but hygiene is improving, right? We are coming to know the various antibiotics and all that. So we are making control over that. But what about the genetic disorders, right? So here, actually, we have to find out, right, what is the cause at the gene level. So in the infectious diseases, we have a lot of control, right? But when genetic disorders, still it requires detailed study, right, at the level of gene. Due to the fact that large number of diseases of all age groups, early childhood, or the middle age, or the old age, right? So in all age groups, right? So the diseases which are occurring in these age groups, right, now known to have genetic background. So in the present situation, right, all the diseases in various age groups ultimately causes going, right, or it has a genetic background. So it has a very big, or the what we can say, right, maximum importance, the genetic considered, right, for the present day diseases, right. So due to that, right, this genetic is very important in this the medical fraternity. We should go in detail about it. We should try to learn, right. So what is occurring exactly at the level of gene, where the hereditary factor, the mystery of life is written, right, the coded in that gene level. And once we able to read that codes, right, so most of the problems, right, diseases, we can solve it. So it is very big event, right, in the as far as considered the genetic is there, right. So with that all, right, background, so once again, right, the genetic plays a very important role, right, in the medicine or the medical fraternity of the present day situation of the various diseases. Almost about 50% of the the first trimesters, right, abortions, they are due to the chromosomal abnormalities. So if we go in detail about the 50%, right, the first trimester abortions, repeated abortions, right. So if one has the repeated abortions in the first trimester, most of the 50% cases of that, if you find out the cause, right, it is at the level of chromosome. So chromosomal abnormalities, it may be at the level of numerical or structural, right? And due to that, right, the chromosomal abnormality, that babies, right, they are not able to survive, so converts into the abortions. So mostly the 50% of such first trimester abortions, right, the cause is, right, into the chromosomal level, right? It may be due to the chromosomal various abnormalities. 2 to 3% newborns, they born, right, with the congenital malformations, and these congenital malformations, they have a genetic origin. So congenital malformations, it may be like cleft palate, cleft lip, developmental or the defective ears, developmental defects in the kidney or heart, all these congenital malformations. If you go in detail, right, why this they are occurring, why these particular organs they are not developing properly, and baby is born with the various malformations, various defects, right, congenital anomalies. again causes going right to the chromosomal level and the genetic origin. So this gene, right, though it is right before birth, right, in the embryonic period or afterward, so it is playing a very important role, right, in the human being. 2% infants with they born right with a single gene disorders. More than half of the childhood with blindness, deafness, mental retardation, it is due to the genetic disorder. More than 5% adult population suffers from the disorders having genetic background. Then presently, right, the cancers, right, tumors, right. So these cancers, they have a strong genetic background. So cancers, right, if you see the previously, right, they're occurring beyond 40 years or beyond the 50 years, but now the age is preponed, right? So we are seeing the cases, right, cancers occurring in mostly 30 or the 20s even also, right? And the reason is, right, again, it will go toward the genes, right? So specifically, right, mutations, what they're occurring, but it is a multifactorial, what we can say effect, right, as a cancer. But one role, right, the genes is playing there, right, gene mutation. It may be the change in the environment, right, environment factors, they're stimulating the genes to make a mutation, and due to that, right, cancer conditions are occurring, and the age is also preponing, right, coming earlier, and all this, right, the multifactorial effects, including the gene, again playing important role in this. That's why this genetic is very important, right, in this the medical education. One should have a detailed knowledge. We should work out what the genes, right, and what exactly in particular diseases or in particular cancers, in particular areas, tissues, cancers, what exactly which gene is concerned, right, for that the cancerous condition or the disease particular condition, and what we can do as a treatment part, right. So here comes the role of the gene therapy, right. We heard that, but toward the western side, right, gene therapy is right more working. In still India, right, it is yet to come, right, it has to work out here. But this gene therapy is a new concept where if you have a detailed study, if you know that particular disease or particular cancer is due to in that particular chromosome number this and this and this genetic that particular gene, right, because in human being, right, human chromosome, right, there are near about 25,000 to 35,000 genes. They're working right in this the human cell, right, uh, and these genes, right, if we have a detailed study, right, so we can label that particular this gene number gene, right, is responsible for this, for example, thyroid cancer. And if we are able to replace it at that particular level, right, so due to that newly gene, right, what we replace there, that particular person will not suffer, right, from that particular disease or cancer. So this is the what we can say revolution in this genetic, right. So in that case, this genetic is playing very important role. After seeing this, the importance of this genetic, right, in medical fraternity, right, or medical education, we'll see some the corners, right, basic important points, right, in this the genetics. So we'll come to the classification of genetic diseases, how they are classified, right. First is a single gene disorder. It is due to the single gene. If the single gene is defective, that also can affect that particular person, that particular baby, and that baby will not, right, be properly developed, and this will label, right, as a single gene disorder. It may be a recessive or dominant. Recessive means, right, so if the two same, right, the recessive parts they come together, then they will express, right. So expression will be right after the combination of two recessive genes there. And for example, right, the diseases we can take, right, quote like this: albinism, autosomal dominant. So dominant means, right, though they are in single number, they are going to express their right characteristics. So autosomal dominant, for example, achondroplasia, where there is a defect in formation of the what we can say, right, the cartilage, and that growth is restricted in that particular person because the growing cartilage is not formed properly there. So due to the single gene disorders, right, so it may affect the development, and it may be autosomal. Autosomal means here, right, one should know that, right, there are 23 pairs of the chromosomes in human being, and out of that 22, they are autosomal, and one is concerned with the sex, right? So that is X and Y chromosome. So rest of this sex link, right, what we can say is autosomal recessive, autosomal dominant. Then concerned with the X chromosome, X linked, right? So that genetic diseases. So if it is dominant, right? So X-linked dominant diseases, for example, vitamin D resistant rickets, and X-linked recessive, right? Example is hemophilia or the color blindness. Means concern with the 22 autosomes, right? So single gene, if it is defective, it can affect disease like this. And if it is concerned with the X-link, right? Whether it's a dominant or recessive, the diseases, the genetic disorders will be like this. So single gene disorders is one type of here, right? Genetic disorders. Then the multifactorial disorders, it is due to the additive effects of the multiple genes. For example, the cleft lip or cleft palate, hypertension, diabetes mellitus. So more than one, right? The gene is defective there, and there is a combined or additive effect, and they can produce, right? This is like this. Then chromosomal abnormality. When chromosomes are not properly formed, right? So due to the increase or decrease in number of chromosomes, right? So as we already said, there are 23 pairs of the chromosomes. So in that 22 are autosomes, and one is the sex chromosome, right? X and Y. So depending on the male and female, in that, if this chromosome number, right, numerical and structural defect, that can also affect or that can create a genetic disorder. So if it is increase or decrease, means more than this, right, what we specified this chromosome numbers. So if it is increase, right, so trisomy 21, right, so what is labeled as the Down syndrome or the Turner syndrome, right, where it is the 1X, right, chromosome is missing, right? So in that case, right, so that particular syndrome is labeled as Turner's syndrome. So that particular baby is female, right? But 1X chromosome is not properly formed. So there is no proper feminine structures formed in that. So that is Turner syndrome. So either decreasing or increasing number also affect the development, and this is the chromosomal abnormality. If there is a minor defects, right, nowadays with the banding technique or the fish, right, technique, we can find out even minor damage, right, or the minor abnormality concerned with the chromosome. Then acquired somatic genetic diseases, somatic cells divisions, mitosis continuously going on. So growth is continuously going on in our body, we know that. So growth means there is cell divisions. So mitosis and meiosis type of cell divisions are there. Meiosis, we know that it occurs in the germ cells, but rest of the cells, right, the mitosis occurring, and this from one cell, two cell, from two cell, four cells, they are occurring there. So cell division is a continuously going process. In that, right, there is a chance of change in structure in the gene mutations, and responsible for various cancers. So acquired means due to maybe exposure to the radiation can do the mutation there, or environmental factors, right? All they can affect on the structure of gene there, and due to that, right, the sudden mutation in that particular genes, right? So they can produce a genetic disease. So acquired somatic genetic diseases will be under this. So this is the classification of genetic diseases, right? Single gene disorder, then multifactorial disorders, chromosomal abnormalities, acquired somatic genetic diseases. All genetic is around the gene, and genes are the informative and the smallest unit that is present in the chromosome. So what we say genetic, so basically the center point is the gene. One should know detail about the gene, where it is located, what are the codings in that particular gene. Then we'll come to know, right, in details about the genetics. These genes are present in chromosomes. Genes are present at particular site or at particular location. That location in genetics, it is labeled as locus. So what do you mean by locus? Locus is nothing but its location or particular position of that particular gene on the chromosomes. And these chromosomes, they're located in the nucleus, right? So everything is located in the that what we can say of the life is present in the nucleus of that particular cell. Chromosomes are very important in genetics. Then something about the cell divisions, as already I have stated, right? Mitosis and meiosis. Mitosis, where the diploid condition is maintained, while meiosis, right, it is the haploid condition, right, from diploid to haploid, means from 23 pairs, right, the 46 chromosomes, right, they are converted or they're reduced to the 23, means diploid to the haploid. Reductional type of the cell division is that meiosis. So it is very important, right? So when gametogenesis occurs at that time, this meiosis, right, which is occurring in the germ cells, right? In case of male, we say as spermatogenesis. In case of female, we say, right, the oogenesis. That time, this reduction is occurring, and if that reduction is not occurred properly, right, so that may affect, right, in the chromosomal numbers or damage to that particular chromosome, and that may convert, right, into the genetic disorders. So cell divisions are very important as considered to the genetics, right? Hence, at the beginning of 20th century, it became known that heredity is controlled by large number of factors. So heredity is controlled by large number of factors. So they are nothing but the genes, and that the genes are located right on the chromosomes. So this is very important discovery, right, in this genetic. So ultimately, the genes which are concerned for the heredity, and they're present in the chromosomes, and chromosomes they're present in the nucleus level of that particular cell. And these cell divisions are very important, mitosis and meiosis. So where this the chromosomes, right, they're separating genes at that time, right? If the proper separation is not there, right, during reduction type or during multiplication, right, duplication, that time it may convert into the genetic disorder. So this is the pictorial explanation, right, regarding this. So this is the cell, in which this is a nucleus, and in the nucleus, right, all this chromosomal material there. So cell division is going on there, right? So mitosis, meiosis is going on. At that time, there are various phases in the cell divisions, right? So at that time, right, these chromosomes, right? So they duplicate, right, when they are forming the new cell, right? So same numbers, right, they're duplicating and then forming the new cell. So that time, right, it is very important at the crucial phase, right? So that should be developed properly. That cell division should be properly there. So you can see here, right, this is the chromosome, right? So in the chromosome that you can see here, right? So these genes are ultimately there. In the chromosome, we are getting the DNA, right? So deoxyribonucleic acid, right? Ultimately, they're forming the genes here, right? These are the various bases, right? So here you get, right? If you see the DNA, right, which is a double-stranded helical structure, right, in which, right, you get one sugar molecule, one get, right, another is phosphoric acid, and third is, right, the base, right? So there are 4 types of bases, and depending on that, various codes are formed, right, in this DNA. So ultimately, this DNA, right, is going toward this chromosome, right, in the nucleus. This is very important area as far as the genetic is considered. So we'll focus right on the human chromosome. What is the structure of it? So already we have seen that chromosome, right, is present in the nucleus. Nucleus of non-dividing cell shows strands of the material. They are called as chromatin. Means the cells are undergoing, right, cell divisions, but when they are not doing cell division, that is resting phase. So non-dividing cell, right, these chromosomes, they are present in the form of a complex network. And this complex network, right, we call as chromatin strands. These chromatins, they are in the form of coiled and extended. When that is non-dividing stage, it is extended, and when it is coming to the dividing type, the phase, right, that interphase or metaphase, that time, right, so they get coiled, right, condense, and they forms the chromosomes there. The chief chemical constituent of chromatin is a DNA, that is deoxyribonucleic acid. Now these 2 words, the heterochromatin and euchromatin, one should know. Heterochromatin means darkly stained, right, or the coiled part or condensed part. So it is darkly stained, right, and this part is area is called as heterochromatin, which is darkly stained in that nucleus. Euchromatin means lightly stained. At that level, that strands, right, they are uncoiled, right, and they are not condensed. Due to that, it is lightly stained. So heterochromatin area, euchromatin area, one should know that, right, the genetic explanation behind it. At time of cell division, chromatin strands condense into the complex structures by helical tight coiling, that is called as chromosomes. So once it gets coiled in a particular manner, right, it forms the chromosomes. And these chromosomes can be seen through the light microscope, because once they condense, the size becomes a little bit more, they are visible, and we can easily observe under the light microscope. This is the picture of the human chromosome, right? One can see here the chromatids here, right? So right half, left half, right? You can see here the joining part, right? So the primary constriction, right? And this is labeled as the centromere, right? These are called as the arms, right, of the chromosome, right? The short arm P, right, long arm, the Q arm. So P arm, Q arm, right? And sometimes we get secondary constrictions, right? So they are called as satellite, right? So in this way, this is the diagram, right, which will explain you, right, the structure of the human chromosome. So two chromatids joining in the center at the level of the centromere, which is a primary constriction, right? And this position, right, where it is primary constriction. So depending on that, human chromosomes, they are classified, right? We will see in the next, right, few slides. So this is the general structure of a human chromosome, right? Chromosome number, the specific, right, for the hello.
Yes, hello.
Can you hear my sound?
Yes, sir. Yes, sir. You are audible and screen is also visible.
Chromosome number, as I have described, right, they are specific for the particular species. In human being, right, there are 46 chromosomes diploid set. So in genetic, it is labeled as 2n. Gametogenesis, it is a reduction in number of chromosomes takes place, right, at the time of this gametogenesis. In case of male, right, we call as the process of formation of sperm, spermatogenesis. So it is a relational type of cell divisions, as we have declared, right? So meiosis is taking place during this gametogenesis, and this haploid set from diploid. So 23 only chromosomes they are maintained there, and this is labeled as the single N. So 2n means diploid set, right? And single n means haploid set. Gametogenesis is formation, the process of formation of gametes. Male gametes is a sperm, so it is a spermatogenesis. Female gamete is ovum, so it is oogenesis. So both during this gametogenesis, right, reductional type of the meiosis, right, meiotic one division is taking place, and it should be happen properly. Otherwise, chromosomal numbers will get disturbed or damaged. That will convert right into genetic disorder. Then autosomes and sex chromosomes. So out of the 46 chromosomes, so 44 they are labeled as autosomes, 22 pairs, and the two chromosomes they are sex chromosomes. Homologous means two chromosomes forming pair. They are of the same, right? Half is coming from mother, half is coming from father. So they are of the same category. And such type, right? It is called as homologous. Heterologous means they are not of the same type. Then sex chromosomes, they are of X and Y. So human female, so it is with the 44, right, plus XX, means two X chromosomes are there, and 44, right, autosomes are there. When human male, right, the genetically we can say, right, so the genotype will be 44 plus X and Y. So in male, this Y chromosome, right, is creating the maleness, right, and here the two X chromosomes, that is female. So genotype of female, right, in genetic we can say 44 plus XX. In case of male, it is 44 plus XY. So 44, there are autosomes which are same in the male and female. When the sex chromosome, right, there is a difference of X and Y here. So that is determining the sex, right? Coming to the size, the human metaphase chromosome, right? It is the largest size and it is near about 5 micron meter. And due to that, right, at this phase, being a large size, we can that is visible, right? We can see it under the microscope, and that is the basic principle in karyotyping, right? So this metaphase chromosome is very important because at this phase, chromosome is large size. It is near about 5 micron meter, and we can observe it visibly, right? See it in the light microscope, right? Very easily. Shape of the human chromosome different during different phases. Interphase means that when it is not dividing, so it is in the non-cell dividing stage. So it is a thin chromatin strands are there. While metaphase, right, thick condensed rod shape. So when it is right in the interphase, it is a thin strands, right, shapes. While it is in metaphase, right, it is condensed and it is rod shape. During the last phases like telophase, right, the anaphase, telophase, that time it may be of J shape or V shape. So there are various shapes, right? But one should remember, right? So when it is non-dividing, that time it forms a complex network of chromatin strands. And during the phases of the cell division, right, metaphase, so it is thickly condensed rod shape chromosome is there. You can see here this right corner, right? So this rod shape right chromosome picture. What is the structure? So already one diagram, right? We have seen there are two chromatids. So they are coming together. Two chromatids are there. One centromere. So that centromere is nothing, right? So it is a junction point of these two chromatids. So two chromatids join. That area particularly also labeled as primary constriction. It is lightly stained area in that human chromosome. So it is a primary constriction, and this is responsible for movement of chromosome during cell division. During the various stages of cell division, right, this centromere is playing very important role. It is moving this chromosome at that point. So it is lightly stained. It is primary constriction human chromosome, or in other word, we can say two chromatids are joined at the centromere level. Then the P arm and Q arm is called. P arm is short arm within that particular human chromosome, and Q arm is a long arm. Then satellite, right, is a secondary constriction. In few chromosomes, right, you may get apart from this primary constriction, the secondary constriction, and it is labeled in this in genetics, right, satellite. This is concerned mostly with the nucleolar organizer area and for nucleolus and its formation. So this satellite is concerned, right, for formation of nucleolus and its formation. So it is a nuclear organizer area, and this is called as secondary constriction, right? In that particular diagram we have seen. Now, how these human chromosomes are classified? As I have said, right, depending on the centromere, right? So based on the centromere, that is primary constriction, human chromosomes can be classified as metacentric, submetacentric, acrocentric, and telocentric. Now, what do you mean by metacentric? When the centromere, that primary constriction, is located in the center, and as it is located in the center, you get two arms, right, which are equal in size. So P arm and Q arm in this metacentric chromosome, it is of more or less equal in length. In submetacentric chromosome, centromere is slightly away from the center, either upper or down, so it is not exactly in the center. So due to that, there is unequal, you get arms, right? So two arms are unequal, and such type of chromosome, right, is called as submetacentric. Then acrocentric, in which centromere is very close to one end, one arm is very short, other arm is very long. So again, very unequal, right? You get distribution of the arms here in this acrocentric chromosome. Then telocentric, in which centromere is present at one end only. So have only one arm. So depending on the position of centromere, right, we can classify our human chromosomes like a metacentric chromosome, submetacentric chromosome, acrocentric chromosome, or telocentric chromosomes. With the diagram, we can explain it. So this is metacentric, in which this primary constriction or centromere, it is more or less in the center. So this P arm and Q arm, they are of equal length. While it is shifting either to this side or this side, so it creates unequal arms, right? Here you get P arm, right? And here you get Q arm. So it is longer and shorter. So just position of this primary constriction, right? Or the centromere is shifting this side. If it is going again, right, more towards the end, right? So it is called as acrocentric chromosome. And in this last picture, right, we can see it is at one end only. So that such type of chromosome is called as telocentric chromosome. So depending on the position of the primary constitution of centromere, we can classify in these 4 subgroups. Then there is a Denver's classification, right? Standard classification called as Denver classification. So in these chromosomes are classified into the 7 groups. So they are grouped in the 7 we can say subgroups as per the descending length. So length of the chromosome is considered here. So descending, right? So longer chromosomes close together, then shorter, then smaller and smaller. Like that, 7 groups. So chromosomes are classified in 7 groups in this Denver classification. These groups, they are labeled as A to G. Means this, that the picture so in this, right, we can see, right, 7 groups A to Z, already I have said, right, and this X chromosome, right, which is placed in C group, and Y chromosome, it is placed in G group. So this is the standard classification, also known as Denver's classification. Then there is a Paris nomenclature, right, the way of classification of these chromosomes. After invention of the banding techniques, more accurate methods for identification of chromosomes came into existence. Long and short arm, they are divided into the 3 regions. So they are labeled as first region, second and third region. So arms are divided here into 3 regions. Then each region divided into the bands. So this is the band technique. This is the basic principle behind the band technique. So these regions, they are divided in arms, they are divided in 3 regions, and in that there are particular bands. So each region divided into the bands, dark band, light band, which can be identified accurately. It helps to detect minor structural abnormalities within the chromosome. So this banding technique, right, is very important to detect very minor defect, right, in the or damage to that particular chromosome. For example, KRIYA syndrome, in which the 5th number, right, the long arm, right, it has a minor defect. It is deletion is there, and due to that, right, that particular baby have a particular symptoms, what we can say, and that is labeled as KRIYA syndrome, in which when the baby is crying, it is looking like the cat sound, right? So that is called as KRIYA. So basic reason is that minor defect in that particular chromosome, and with this band technique, right, or the fish technique, we can detect it, right, what chromosome, defect or damage. Diagrammatically, we can see here, right? So these are the arms, P arm, Q arm. So they are divided into the 3 regions, right? 1, 2. Here you can see this 1, 2. Then in that, right, 1, 2, 3, right? So these regions, and in that we can see, right, these bands. These are the particular bands, dark band, light band, all that. And these bands, right, we are more accurate, right, to diagnose the minor defects in these chromosomes, right, with this banding various techniques. There are various types of G banding and all that, right? So we are not going in detail, but banding techniques is a separate area in this, right, genetic, with which we can detect, right, the minor defects, right, in the chromosomal level. Once we have seen all this, right, the basic information regarding the chromosome, human chromosome, so how this analysis of chromosome can be done? This chromosome analysis is required, right, in certain clinical conditions. So where it requires such, the clinical conditions are congenital malformations, mental retardation, repeated abortions, sex determination, and prenatal diagnosis. So various congenital malformations, for example, if the baby, right, is with the cleft lip, so in such case, it is indicated that we should do the chromosomal level study. If the mental development, right, various the developmental milestones, they are not in proper way, so baby is not mentally properly developed. If you feel like that, that time we can go right in the study of chromosome analysis. Or repeated abortions, as I have said, 50% of first trimester abortions, the reason is in the genetic, right? So in that time also we can do the chromosomal study of that particular mother. Or the sex determination, right, where we want, right, to determine whether this particular baby, right, is a male or female, the borderline cases. So where we want to determine the sex, right, genetical sex. In that case, we can do chromosomal analysis. We can search out for the either X chromosome, two X chromosomes are present, or X and Y. So this is mostly for the borderline cases, right? So here it is needed. That time we can do a chromosome analysis. Prenatal diagnosis. So in case of, right, suspected, right, the developmental defects, right, in that particular developing baby, if we are suspected, that time we can do a prenatal diagnosis by chromosome analysis. We can do a chromosomal study, and that is called as analysis. Now, when we are studying this chromosome analysis, right, one should know, right, about the karyotype. What do we mean by karyotype? Right, karyotype is nothing but the chromosomal constitution of an individual. What is a chromosomal, what we can say, configuration in that particular individual is called as karyotype. Photomicrographs of an individual's chromosome arranged in standard manner. So this is, right, overall is called as karyotype, and the process is called as karyotyping. When it is explained, right, with the help of diagram that represent that particular karyotype, so it is labeled as ideogram. In the genetic, it is called as ideogram. So one should know that during chromosome analysis, right, where it is indicated, what are the indications, then words like explanation of the words like karyotype and ideogram, and the process is called as karyotyping. In short, we'll see, right, how the chromosomal analysis by karyotyping is done. So this procedure, which is labeled as karyotype, right? So metaphase chromosome of somatic cell is obtained and photomicrograph. As we have seen the importance of this metaphase chromosome, because it is highly condensed, it is rod shape, and we can see it easily. So visibility is clear at the level of metaphase chromosome level. So in this karyotyping, this metaphase chromosome is very important, right? Individual chromosomes are cut and arranged according to the standard classification, Danvers classification. Rapidly dividing cells are used in this karyotyping. So what are the sources of this rapidly dividing cells in this karyotyping? They are the lymphocytes from the peripheral blood, peripheral venous blood we can collect in that lymphocytes. It is a major source, right, we can utilize, which is rapidly dividing there, right, for the karyotyping, and it is commonly used. Then fibroblast from the skin, bone marrow cells, chorionic villi, amniotic fluid cells. So these are the various sources. We can take the cells and we can do a chromosomal analysis, right? 5 ml of the blood is collected under the sterile condition. All antiseptic precautions are taken. Peripheral venous blood we can collect. Then heparin is added, right, which is for to prevent the clotting as anti-clotting agent. We have to add heparin in that. Lymphocytes are separated off from the RBC, right, by the centrifugal force. Then white cell suspension kept in cultural vial, right? So culture vial, which consists of the media and fetal calf serum, which is acting as a nutrient agent there. So that in culture vial, right, we have to keep these white cells, lymphocytes. Then phytohemagglutinin is added because this agent, it stimulates the cell division, because we have to do it rapidly cell division so that we can catch that the metaphase chromosome. So we have to stimulate, right, there the cell division rate, and this is done by the phytohemagglutinin. Also the antibiotics, they are added there to prevent the infection, any sort of infection we don't want to, right, create in between. All this, the material kept in incubator for the 3 days, near about approximately 72 hours at the 37 degree centigrade. Then a very important agent called as colchicine is added in this. This colchicine, right, it prevents, right, the spindle formation. The spindles, they are the pathway, right, to move the chromosomes, right, from the equator level. So this colchicine agent does not allow to form the spindle, so that metaphase chromosome is arrested there. So that particular cell division, right, at metaphase, it is arrested. So we get metaphase chromosome there, right? So this colchicine agent, after the 72 hours, right, we have to add, and this is very, very important time here, right, or important phase in this karyotyping, which prevent the spindle formation and that arrest the cell division at metaphase level. At metaphase, chromosomes are maximally condensed and they're visible easily. After 2 hours, lymphocytes are separated by centrifuge. These cells treated with hypotonic solution. This is very important time. So get swell due to the hypotonic solution, right? Cells get swell and chromosomes, they're separate. Then these cells, they get fixed in the glacial acetic acid and methanol. Cells suspended in fixative are dropped. Now here we have to do a mechanical rupture of that cell. So these cells, red material, drop on the chilled slides from the height. So cell wall get ruptured and chromosomes can spread well. So chromosomes get spread over that slide. Then we have to stain it and we have to do a micro photographs. Then individual chromosome, right, after seeing the light microscope, we can separate out it, we can cut it individually, and as per the length, right, height, we can arrange it. Thus karyotype is obtained. So karyotyping is a procedure by which, right, the chromosomal configuration in that particular individual is done. And in that, at the end, we have to do photomicrograph. We have to arrange all these chromosomes as per the Denver's classification. We have to arrange in 7 group, and then we have to work out, right? We have to see it, right? For search for the X chromosome or Y chromosome. Now this is, right, the karyotype, right? So you can say, right, this diagram in which, right, all chromosomes, they are identified, they are cut, then photomicrograph, and at the end, right. So this is the way of presentation, right, in karyotype. At the end, right, they are showing very important 6 chromosomes here. This is the X chromosome, this is the Y chromosome. So this is a karyotype of a normal male. You can see all the chromosomes here, autosomes, right? You can see here X and Y configuration. So this belongs to the normal male. This karyotype, you can see, right, we find out here 2 X chromosomes. There is absence of Y chromosome, means this is a female. So this is a karyotype of a normal female. If there is a borderline case, right, coming in OPD, so we suspect, right, what sex is there, right? So genetically we have to find out. In such case, we can find out, take the female blood, do all this karyotyping procedure, and at the end, right, if you are able to find out there are 2 presence of 2 X chromosome, means genetically that particular baby is female. And if we got, right, 1 X and 1 Y, means that genetically that baby is male type. Like that, this karyotype is important during the sex determination. Along with this, right, one should know that, right, what is a sex chromatin or bar body. The female interface, right, nucleus shows a dark stained chromatin mass attached to one side of the nuclear membrane. The dark stained body is also called as bar body, present only in females, they're absent in males. So this is very important. Why it is? Because there are 2 X chromosomes in females. One is inactive. Of the males, right, X chromosome. So, right, so this sex chromatin, they're important one to find out whether it's a male or female. The female cells show sex is more accurate. The Lyons hypothesis, 1962, right? Lyons stated this, right, on this the bar technique, right? Hypothesis. Chromosomes -1 equal to number of the bar body. Example, the female have 2 chromosomes. In case of normal male, right? So there is only 1 X chromosome. -1 equal to 0 bar bodies, right? So these bar bodies, right, they're also giving some idea, right, about this, either it's a male or female, but rare, right, like a Turner syndrome, right, where 1 X is missing, that time we are giving, right, little bit the deviated information. Or in case of Klinefelter syndrome, right, where it is 47 XXY, so there are 2 X chromosomes -1. By this Lyons hypothesis, right, you are getting 1 bar body. So these are the bar bodies here. You can see, right, the nuclear cell wall membrane here, right, you can see this is the darkly stained area. Inactive X chromosome is there, right, that is called as bar body, right, or darkly stained area here. So sometimes it is labeled as drumstick appearance also. Now, molecular genetic is more advanced genetic, right? What we have seen cytogenetics, then again more detail to that, right? The molecular genetic, which is a more advanced genetic study, includes study at the molecular level. Genes are present on chromosomes, responsible for inheritance of the characters. They are present in nucleus and mitochondria. Chemically, chromosomes are composed of proteins and nucleic acid. Nucleic acid is responsible for inheritance. Nucleic acids, there are two types, DNA, RNA. So in DNA, right, labeled as the deoxyribonucleic acid, and RNA as a ribonucleic acid. And the DNA is basically responsible for inheritance, right, or the concern for the genes, right? DNA is mainly found in chromosomes and mitochondria. Genes are made up of DNA. DNA, by definition, we know that it is a double-stranded helical structure composed of one sugar molecule, one phosphoric acid molecule, one nitrogenous base. Right? So there are four types of these nitrogenous bases: adenine, labeled as A; thymine, labeled as T; cytosine, C; guanine, G. By replication, new strand of DNA form during the cell division. Gene function to direct the synthesis of protein. Basically, gene guides to develop the protein, synthesis of protein. And this is the main base, right? Protein is involved, right? So protein is formed by the 20 different amino acids, and they are forming proteins. And basically, proteins are involved in formation of various structures, right? Like enzymes, hormones, and all that. So basic reason, right? Control is done by this gene level. A sequence of 3 bases on DNA strand codes for one amino acid, that is called a genetic code. So this genetic code is very important. Thus, genetic codes are present in genes. By transcription, there is a special procedure called as transcription. These triplet codes are transferred from DNA to messenger RNA, and this is called as codon. So once the DNA forms this code, it is transferred to the messenger RNA. It is going away from this the cell wall nuclear level membrane, coming to the cytoplasm with along with this codon. If heritable changes occurs in the structure of the gene called as gene mutation. So if there are certain changes occurs in this gene, right? So that can be transferred from one generation to the next generation, and this is called a gene mutation, or that particular point, right? Mutation. If we are able to find out, it is called as point mutation. So something about the chromosomal abnormalities. It may be of numerical or structural. Chromosomal, this numerical chromosomal abnormalities. Numerical means number wise. If the number is deviated from its normal, right, 46 chromosome, the defect is going to form, like either increase or decrease chromosomal number. So Down syndrome, when there is a trisomy, right, one excessive chromosome at the level of 21, so trisomy 21. Then Patau syndrome, when there is trisomy 13. The names of that scientist who work on that, right, particular genetic genotype here found, the names are given of that scientist to that syndrome. So Edward syndrome, when there is trisomy of 18 number chromosome. Turner syndrome, when X chromosome, right, is missing. So decrease in number here, right? Here it is increasing, here it is decreasing. Here is also increase, right? You get double X chromosome, Klinefelter syndrome. Or super females, when there is apart from 2X chromosome, one additional X chromosome is there, right? So these are all numerical chromosomal abnormalities. We are not going in detail, right? So only the name wise, right, we should know. Structural abnormalities, when the particular structure of that chromosome, right, is damaged, right? So there are various methods, right, by deletion or by inversion, the ring chromosome, right, isochromosome or translocation. So structural abnormalities can occur in that particular chromosome, and that may affect into the abnormal baby. Genetic also plays an important role, right, in the prenatal diagnosis of various genetic disorders. Indications: family history of genetic disorder. That time we should go, right? The particular couple should go for the prenatal diagnosis or genetic study. Advanced maternal age. All the mothers beyond 35 years, because in this the meiosis is most of time, right, the abnormal type occurring, chances of occurring their abnormal the meiotic one cell division. Then family history of neural tube defects, such couples also can go, right, for the prenatal diagnosis. The previous child with chromosomal defects, yes, this is also sensitive area, one can go, right, in the prenatal diagnosis. There are various techniques which are involved in this prenatal diagnosis. By doing amniocentesis, right, we can take the amniotic fluid in that particular cells are present, right, from that baby, and we can study the nuclear level of that particular cell, and we can able to know, right, so what defect is going to occur in that or normal baby. Then chorionic villi biopsy. So we can take a part of that villi, right? So that also will indicate, right, when you study, we'll get the particular embryological defects in that baby. Fetoscopy, right? You can do a fetoscopy. You can study that fetus, and you can come to know, right? So what abnormality is going to there. By ultrasonography also, we can detect the various malformations, and then we can prefer, right? We can go for the prenatal diagnostic test. Maternal serum screening for the AFP, right? The alpha fetoprotein, right? So this alpha fetoprotein level, if it is excessive, it indicates the neural tube defects. So that is also indicator, right? So we can do a maternal serum screening, right, for this level of AFP, right? There are the various techniques where we can do a prenatal diagnosis, right? Modern and advanced techniques in genetic, as we have seen, right, genetic plays a very expansive role, right, in this medical fraternity. Nowadays, every branch is, right, developing very, or it is going in details, right, as every disease, right, the root or the cause is going toward the genetic, right. So we have to study in detail. So advanced research are going on, right, in this genetic. So these are the few names, right, concerned with this advanced techniques. We are not going in detail, but one should know about this, right? Recombination DNA technology, DNA cloning, nucleic acid probes, DNA sequencing, DNA fingerprinting, human cloning, human genome projects. Recombination DNA technology, right, in which the particular DNA fragments, right, so it's a duplication is formed, right, with the help of the in vitro or in vivo, right, the formation there. In particular, that fragments of DNA, right, they are prepared. Then DNA cloning, particular type of codes, right, we are doing repeated production of it, right, same type, which is called as DNA cloning. The nucleic acid probes, so particular duplicates, right, of that particular codes which are required for synthesis of protein, they are also prepared. Nowadays, right, probes are also ready there, right, and we can replace, we can do treatment with that particular probes. DNA sequencing, right, sequencing of that particular the base, right, over that the double stranded structure of the DNA, right. So that sequencing also, right, we can study in detail, right, we can or that advanced study is going on over that. Something with the DNA fingerprinting also, right. So this is mostly in case of proving, right, that particular paternity, right, that particular baby is belonging to that mother or father, right. So we can take that the whatever the material, right, from that mother or father and from that particular baby, right, and we have to do the fingerprinting, right, we have to match. If it is more than 50% matching, that particular baby is belonging to that mother, or that father is genetically declared, right, he is the father of that particular child. So in crime also, right, this DNA fingerprinting is very the useful technique, right. Then human cloning, right, cloning means forming the same that genome, right, same copies. Now this cloning is banned, right, all over the world, right, because it has many various disadvantages, right, more disadvantages, so it is banned. So this human cloning also technique in which, right, the same copies, right, we can produce from that particular course, right. So but that is this we can utilize, right, for the other diseases, but we cannot clone, right, of a particular human, right, being, so which is banned technique. Then human genome projects, right, so in which, right, as we have seen, there are about 25,000 to 35,000 genes, right. So study of all these genes, right, which gene is required for what control he is doing, right, all this is studied in this project, right, which is called as human genome project. These are the advanced corners, right, concerned with this genetic. We are not going detail, but just briefing over that, right, what they are concerned with. In this way, genetic is assuming, right, an increasingly important role in medical education. As a result, teaching of genetics should also be increased, right, proportionally, to ensure that the future physicians will be able to take advantage of new genetic technology and to understand the associated ethical, legal, and social issues. At present, this genetic, right, if you see toward the genetic, it is the most important but mostly neglected branch. Exposure of undergraduate, postgraduate students to the genetic is very less. Only basic topics, not in details, they are exposed to the students. Also, exam point of view, very less contribution in theory and practical aspect. which promote to neglect it. No separate subjects in curriculum. At present, most of the places genetic lab setups are not at full stage. So present at medical college or hospital level. So no laboratory exposure. So interest does not increases. No more number of training centers as experts are very less. No courses available freely, right, or known to all the common people, right, regarding the genetics. Mostly all worried about the jobs and futures, right, in genetic, as it is not well known, right, to the common man or the in this medical fraternity. Overall of this effect in practice with the less knowledge, less exposure, right, when the student, right, after taking degree, right, he goes in society as a practitioner, right, that time again he feels, right, some darkness, right, what to do of such genetic diseases, right, where to refer and how to treat, right, how to do genetic counseling and all that, what is the ultimate solution for this. So there is a little bit questionable mark, right, in front of all practitioners, right. So here also that area we have to clarify, right. Nowadays almost all diseases directing toward the genetic background. So genetic have an expensive role in this medical education. Now, along with this background, right, this is my personal opinion, right, what we can do. Exposure of genetic to the medical student should be increased, right, from the first year to the last year. Increase more contribution in theory and practical pattern. In curriculum, can keep separate paper of genetic, it will be very good, right, so student will give more weightage to it, they can learn in more details. Develop and maintain genetic labs. So it will be very good, right? Genetic lab is there in particular hospitals, right? So student can go at any time, they will have exposure of the various practical procedures. Keep posting of interns compulsory in this labs so that they can learn actual work and interest will increase, right, toward the genetic. Arrange more genetic workshops, CMEs, conferences like this today's national conference. So to attract the new budding doctors and practitioners and students toward the genetics. Promote research work, right? That research work is going at higher level, but a common student, right, should also involve in this research of the genetics, provided, right, infrastructure is there to that student, he may definitely he will go towards or attract toward the various researches. So this is my personal opinion, what we can do, right, to improve or to do more work, right, in the genetics, right. With this, right, I conclude, right, my this topic. I'm sorry that I extended, right, 10 to 15 minutes more as per the schedule. It is now 12:15, right, expected to finish by 12. But anyhow, topic is very extensive, right. The genetic is no doubt playing very important role in this present medical education. What we talk
Thank you very much, sir. Dr. Sudhir Pawar, sir. Dr. Sudhir Pawar, sir, has delivered a very informative lecture on genetics, a role of genetics in medical education, various concepts into the genetics, which has been elaborated by Dr. Pawar, sir. Our first is the importance of genetics in the medicine, karyotyping, Lyon's hypothesis, the inverse classification is also explored by the sir. Various genetic component associated with the various disorders like cancers and other basis, genetic basis regarding other disorders has also been very nicely expressed by the Dr. Pawar sir.
was
Advanced
given chance to me
regarding the
to
genetics
explain.
like chromosomal analysis and their benefits, advantages are nicely displayed by the Dr. Pawar sir. And I am very much thankful to Dr. Pawar sir for this informative session. Thank you very much, sir.
Thank you, sir. It's my pleasure. It's my pleasure to explain, right, as you have given opportunity to explain, right, to talk something about the genetic. So I'm very much thankful to the organizers to the head of the Department of Physiology, Syed sir, and Kanab sir, and all the respective dignitaries, right? So I'm very helpful. I'm very thankful. And in future also, right? If such workshops going on, right? I'll be happy to share. And once again, right? Thanks to all for giving this opportunity. Within short time, right? As I get the message from you, so but within short time, right? We'll be able to make all these preparations, right? Also from your side and my side. So it was a wonderful experience, right? To contribute or to talk.
Moving on towards the next session of today's webinar to be guided by Dr. Aniket Shirvansar. Honorable Dr.
Is there
Aniket
is there no time for question and answers?
Assistant Professor in Department of Kriya Sharir of G.J. Patel Institute of Ayurvedic Research, Anand Gujarat. He has completed his BMS MD in Ayurvedic Research and International General Moving on towards next session of today's webinar guided by Dr. Aniket Shilvan sir. Honorable Dr. Aniket Shilvan sir is working as Assistant Professor in Department of Kriya Shari, G.J. Patel Institute of Ayurvedic Research, Anand, Gujarat. He has completed his BMS MD in Kriya Shari from Government Ayurvedic College, Nagpur. His teaching experience is 7 years. He actively participated in more than 50 various seminars, CME, conferences, workshops, and teacher training programs at national and international level. He has published 3 books, An Analogy of Jatharagni and Psyche in Ayurvedic and Modern Parlance, Indriya Shari, Understanding of Deha Prakriti Made Easy. He has published about 35 research papers and articles are in national and international journals, about 8 paper and poster presentations in various seminars. He has been credited for resource person for Aish Guru web portal, guest speaker for various concepts of Ayurveda and modern science. I would like to invite Dr. Aniket Shilvan sir on this platform as a speaker of this session. His topic is Prakriti and Genetics. Sir, please continue with your topic.
Yeah, sure. Thank you. First of all, am I audible? Am I audible?
Yes, sir.
Yeah. First of all, I would like to thank you to all the organizers, teaching staff who are directly and indirectly involved for arrangement of today's webinar and for the very beautiful topic that is genetics, its role in Ayurveda and medical sciences, modern medical sciences. For me today, the topic given was Prakriti and genetics. So we will be exploring regarding the formation of Prakriti and its genetic point of view from modern as well as Ayurvedic point of view at a single platform. So let me share the screen first. Is the screen visible, sir?
Yes, sir. Yes, sir.
So we'll be starting with our today's first topic, that is Prakriti and genetics. And our key coverage areas are, we will be having in detail about the Prakriti in Ayurveda. We will be exploring the concept of Dosh Anushayita, Dosh Utkatatva. We will be looking forward for factors which are essential for formation of Prakriti. We will be looking for types of Prakriti, the role of Garbhakar Bhavas, Shad Garbhakar Bhavas are there. We'll be having some modern tests regarding genes, genotypes, phenotypes, and alleles are there. Basically, modern regarding modern types, already the Dr. Pawar sir has explained regarding genetics and its role, but we'll be having just a simple touch regarding genes, genotype, phenotype, allele.
Then after we will looking for Ayurgenomics is there, Nutrigenomics is there, epigenetics is there, and the last importance of Prakriti, which will be covering the scope of Prakriti or scope of research regarding Prakriti as well. So to start with very basic, when we start introducing Prakriti to first year students, we need to explore this quotation basically that is called as Prakritistu Swabhav. Now from the word itself, it starts regarding phenotype. Whatever phenotypic or traits which are expressing or manifesting, which represents a set of behavior characteristics of ourselves, that is nothing but Swabhav. So we are having the same quotations in our ancient science also, that is Prakritistu Swabhav is there, Prakritistu Swabhav Dharma is there, Prakritistu Sharir Swarupam is there. So which includes all the four aspects, that is morphological is there, then after psychological is there, then after behavioral is there, which includes spiritual as well. So we need to understand this Prakriti something beyond not only genetics, right? So there are a lot of synonyms as you can go through this slide. Prakriti, Sri, Swabhav, Mijaz, Vikas, Parampara, Udgam Sthal, Nya, Vaisheshya, Gata, and the other things. From Western schools of medicine, they can understand or they get to know the Prakriti in terms of personality is there, nature is there, behavior is there, temperament is there, constitution is there, rather somatotype is there, and lastly, most familiar word, that is triads are there, right? So, as I had to explain in the very earlier slide, that is, it is just a presentation of or expression of oneself in terms of morphological is there, physiological, psychological, and social aspects, which together combines the swabhava of that particular individual. So basically, Prakriti to Swabhav indicates the phenotypic as well as genotypic expression. Both the things are there, right? So again, now we will move forward with definition of Prakriti. When we come to definition of Prakriti, it is said that Prakriti Nam Janma Marnantaral Bhavini. Now it starts from the conception stage, right? So it is nothing but a set of behavior characteristics which is represented by or manifested by an individual, but it starts from the conception stage, which dwells throughout the embryological stage as well. By the virtue of doshas, I would like to say here, doshas which are nothing but the properties of guna, group of properties, right? So which are possible only with the help of doshas, that too, which should be in the physiological normal limits. I would like to highlight the last word here, that is Nirvikarini Doshtiti. So it represents nothing but the counterbalancing act of the properties, that is nothing but guna, right? By the help of, by the virtue of dosha. So when we nomenclate that this is dosha sthiti, we need to understand this is nothing but the counterbalancing strategy, counterbalancing act of gunas, which try to balance each other. They do not go on destroying each other. They try to counterbalance. They try to maintain and sustain the functional integrity which remains throughout the lifespan. Now this is nothing but the genotypic expression of that particular individual, and this is what we call as Prakriti, right? So the definition which is shown in the Rasa Vaisheshik Sutra is having some extended view beyond just a definition of a Prakriti, right? So again, now we will move towards the next topic, that is Dosh Anushayita, what we had seen in the key coverage area. I would like to highlight here again the gunas of doshas, which are nothing but the Prakriti. When we talk about Prakriti, it is nothing but the healthy and competitive conjugation of doshas. Again, doshas, wherever we see doshas, we need to understand these are nothing but the group of properties, right? So the group of properties having healthy and competitive conjugation in between each other at the time of Garbha Pranti are responsible for development of Prakriti. And this is what is being carried from generation to generation. Now, when we talk about carrying forward, when we talk about generation to generation, we need to understand this word, that is Anushaitam, right? So Dosh Anushaitam, the group of properties or the constitutional makeup which is formed at the Garbha Pranti is being transformed from generation to generation, and that represents the characteristic of that particular individual. So when we talk about all these gunas, we need to understand the proportion, right? So proportion of dosha is nothing but the proportionate dominance of guna. This is the concept of dosha utkatatva, right? So when we talk about dosha utkatatva, we need to understand the proportionate dominance of guna in such a way that they are not trying to destroy each other, but they are trying to stabilize each other by the help of each other, and this is what nothing but the formation of Prakriti is there, right? Now we will explore some more beyond this dosh anushayita. As you all know, the definition of dosha, right? In the very first year, many of the students learn dosh means dushyanti iti dosha, but we need to understand definition of dosha, that is prakriti arambhakatvam saiti swatantrena dushti kartutvam doshatvam, right? So the very first phrase here is prakriti arambhakatvam. We need to understand something beyond this word Prakriti. So when we talk about dosha Prakriti Arambhakatvam, they are not talking only about the formation of Prakriti or chromatotypes, right? They are talking about the normalcy. So normalcy in terms of any particular function, normalcy in terms of any particular regulation of any activity. So that accounts to, that is regulated by dosha. This is the thing, but the first phrase which accounts to the definition of dosha, that is Prakriti Arambhakatvam, right? Now, we are talking dosha are responsible, or doshas are the physiological identity, right? So by the virtue of guna only they are possible to do that. So we need to understand the importance of guna again here. So gunas of doshas are nothing but the blueprint of dosha, which marks their presence now on Garbhakar Bhava. The reference coming from Sharir chapter 3. So the guna of doshas tries to mark their presence on the Shada Garbhakar Bhava. We will try to explore some of them very importantly. So first one is, that is Shukra Shodit, right? Which are nothing but the gametes. So when the gunas try to mark their presence at the gametic level, they are trying to create the genotypic and phenotypic expression at that level, right? When we talk about sattva, this guna at sattva are trying to develop a psychological understanding. Now this psychological understanding is not only of a complete human being, right? We are talking about a psychological adaptability of any particular cell which is facing any particular favorable or unfavorable environment, right? So gunas of doshas mark their presence on sattva also. Next is rasa, most important. The nutritional ability of a particular cell is also decided by the dominance of that dosha, right? And lastly, the satmya, adaptability, or we can say assimilation, right? Property of any particular cell, which is later on accepted wholeheartedly by that particular individual. This is only possible because of the dominance of the dosha. So this is what we need to understand under the concept of dosha anushayita, right? Again, moving forward, we have another definition coming from Shushru, that is Shukra Shonat Sanyoge, Yobhavet Dosha Utkata, right? So wherever we found this word Dosha Utkata, this Utkata is nothing but explaining the physiological limit. The same is quoted in the commentary also, that is Prabhavastito na Prakupita. Means dosh within their physiological normal limit try to dominate each other and represent the maximum characteristics of oneself. And this is nothing but the phenotypic expression of that particular individual, which is expressed as triad, as well as at genetic level, which forms the genotype. So this is Prakriti in Ayurveda, the base of physiological expressions of a particular individual, right? Now, dosh utkatvata, we will go into detail. I would like to quote here, when we talk about guna, frequently we are talking about dosha, frequently we are talking about guna. So we need to understand that dosha utkatatva means we are talking about the dominance of dosha, dominance of guna here, right? So the predominance of guna, which is expressed as dosha utkatatva, is responsible for triad expression, right? So this triad expression is basically founded at both the levels, that is genotypically and phenotypically. What I would like to tell here is the dosha, the gunas of that particular dosha, when they try to counterbalance each other, they are in a balanced state. So this is what we call as a samadosha prakriti, and this can be easily studied under the concept of prakriti samasamved siddhant, right? So we are having prakriti samasamved siddhant, which is only possible. Samadosha prakriti is only possible because of this prakriti samasamved siddhant. But when we talk about dosha utkatatva, any of that particular dosha, by the respect of any of these particular gunas which are explained here, right, mentioned here, try to dominate the other gunas. So when we talk about this domination, we are talking about the Vikruti Visham Samvad. Even though they are counterbalancing each other, but they do not go destroying each other. And this is the situation where Vatadi Prakriti is being formed, right? So Vatadi Prakriti, Pittadi Prakriti, Kapadi Prakriti, when we talk about this, these are nothing but the outcome of Vikruti Visham Samved Siddhant. When we talk about Prakriti Samved, we are having only Samadosha Prakriti, right? So on the contrary, we can also see that this is happening at genotypic level, genes level. So genes are basically nothing but structures which influence the expression of triad. So whatever we are facing the situation at genotypic level, genes level, the guna tries to manifest its expression at the time of triad, right? On the opposite, we can see that suppression of the opposite dosha, means guna only, leads to recessiveness of that particular thing, and where the triad is being suppressed. Now this suppression of triad happens again at both the levels, genotypically as well as phenotypically. This is nothing but dosha utkatatva concept of prakriti. Again, I would like to explain some more about dosha utkatattva. We all know that Shukra and Shonit. So we are talking about Shukra and Artha. One is Saumya, one is Agneya, right? So wherever we are talking about Utkatattva of dosha, Utkatattva of any particular guna, we also know that Shukra and Artha are having typical some gunas there, right? So these gunas try to manifest their presence by expressing dominantly about any particular thing, either Shukra or Artha. So they don't go, Shukra and Shonit, they don't try to destroy each other. Rather, we talk about the amalgamation, right? So everybody is familiar with this word amalgamation, right? Conjugation. So conjugation or amalgamation of Shukra and Shoni by the presence or by the dominance of this guna, which is nothing but dosha utkatattva, is something new we need to study under this concept of dosha utkatattva of Prakriti, right? Again, we need to understand some more beyond dosha utkatattva. When we talk about dosha and guna, as I told you earlier, the dosha and guna don't nullify the effect of each other. We need to understand this very carefully because they don't go on destroying. Rather, they counterbalance each other, right? By sharing their attributes, and that too in a proportionate manner, right? Obviously, when we talk about guna, every gun is having an elemental disposition, right? Because when we study Panchabhutik Siddhant, there is a first quotation, Sarvadravya Panchabhutik Asminarthi. So every gun is again made up of Panchamahabhut, right? So the proportionate elemental disposition of that particular guna tries to balance the other one. And in this way, they don't go destroying each other. And this is the concept we learn under this quotation when we talk about Vishajato Yatha Kitona Vishena Vipadyate. It means this is nothing but the harmless nature of Prakriti. When we talk, for example, a poison of a snake, right? It is poisonous for us, but it is not poison for that particular species, snake species. So this is the harmless nature of Prakriti we need to understand. So Vata Prakriti, for example, of any person is harmful when it goes beyond the physiological limits. But within the physiological limits, it is neither destroying its own parameters. So this is the harmless nature of Prakriti we need to understand, and this is possible only because of the dosha utkatattva concept, right? Again, we will learn something more about dosha utkatattva. Dosha utkatattva, when we talk about dominance of dosha, dominance of guna, this is nothing but we all are coming from the 12th stream where we had learned something about maths also. So I would like to tell this is nothing but a chance of occurrence, right? So this is nothing but the chance of occurrence, or rather, this is chance of probability of a likelihood of an event, right? Occurrence of an event or likelihood of an event is talking about dosha utkatattva. As we know, every guna from each dosha is liable to undergo number of permutation combinations with one another, right? So this happens at the time of conception level. This happens at the time of embryological gestational level, right? Throughout the gestation, it happens. So very first, at the time of conception, when it happens, this is trying to create a genotypic ratio. The proportionate relation of all the gunas is trying to create the genotypic ratio. And whenever it is expressing itself, it is possible only because of the phenotypic ratio. And this can be studied with the help of Punnett square, right? So this particular image is representing the Punnett square. It is an experimental research. If anybody is interested, we can go through this. We can try to elaborate, as I had elaborated here. For example, we are discussing about the Varna, right? So we are having typical Varna of Vata Prakriti, that is Dusar, Varna of Pitta Prakriti, Avdat plus Pittang is there, a Varna of Kapha, Avdat plus Shukla is there, right? But if we try to manipulate this as per, or try to plot this as per the Punnett square, we may see that wherever this capital V, that indicates dominant nature, right? So dominant nature of that particular guna. So which guna is responsible for the Varna? In case of Vata, Ruksha guna is dominant. In case of Pitta, where there is Ushna plus Pita guna is there, right? In case of Kapha, there is Snigdha plus Shita guna is there, right? Shukla guna is there, sorry. So these all dominance of that particular guna are responsible for development of Varna. So in this case, you may see when V, capital V stands there, we may see that dominant nature of this, and this represents the triad of Vata Prakriti, that is Tusar Varna. Here, Avdat plus Pittanga, Pitta Prakriti. Avdat plus Shuklanga, this is Kapha Prakriti. So this is just an experimental example. We can study all the other characteristics or features or triads of Prakriti with the help of Punnett square, right? So this is nothing but the dosha utkatattva, something beyond our textual sciences, right? So this is all about the dosha utkatattva concept of Prakriti. Now we will see formation of Prakriti. These quotations we had learned earlier, Shukra Shonit Sanyoga Yoga Bhaveto Shukataha. So Utkata indicates the Swabhav Sthituna Prakupita, right? So this comes under the concept of, again, we are having a concept in under the physiology syllabus, that is Prakrut and Vaikrut dosha, right? So the Prakrut dosha, which tries to balance each and every guna by the Prakruti Sama Samved Siddhant, are responsible for formation of Samadosha Prakruti. When we talk about Vaikruta, they are responsible for Utkatata. So Utkatata of any particular dosha represents the other 6 different Prakrutis are there, right? We had learned about Doshanu Shaita also. So moving forward, we will be discussing about the factors which are responsible for formation of Prakriti, right? So as you all know, in Charak Vimanstan chapter 8, it has been discussed clearly. There are 4 different factors which are essential for formation of Prakriti, right? The very first, Shukra Shonit Garbhashaya Prakriti. Second, that is Kala Garbhashaya Prakriti. Third, that is Matur Ahar Vihar Prakriti. And the last one, that is Mahabhut Vikar Prakriti. We will go in detail of each and every factor now. So Shukra Shonit Prakriti is nothing but the male and female gametes. We need to understand the purity or pure nature of this male and female gametes. Kala Garbhashaya Prakriti, it itself indicates regarding time, age, season, as well as uterine factor. The condition of uterus is there. Matur Ahar Vihar Prakriti, where it indicates dietary and daily routine practices of mother throughout the gestation period again. And Mahabhut Vikar Prakriti, where whatever we consume, whatever we face or interact with, these are nothing but trying to influence our body constitution, right? So this comes under the metaphysical and elemental factors. So altogether, these are factors for formation of Prakriti. Along with this, I would like to also quote that there are certain factors which represent, which influence the formation of Prakriti. These are nothing but Garbha Vrittikar Bhava. These are 6 in number: Matruj Bhav, Pitruj Bhav, Atmaj Bhav, Satmej Bhav, Rasaj Bhav, and Satvaj Bhav, which are indicated in Charak Sharir 3. So altogether, they are having an influence for the formation of Prakriti. So this is again, we need to understand under the broad umbrella term for factors for formation of Prakriti, right? So first, Shukra Shonit Prakriti. So Shukra Shonit Prakriti, when we need to understand the gametes, we are talking about the Matruj Bhav. So it is, these are certain Matruj Bhav which are developed from the Matruj element. That is, we are talking about the Artha, right? So female gamete, which is basically Agneya in nature. When we talk about the Shuddha, means pure Agneya, pure Artha must be there. We are having a quotation indicating the purity of that particular Artha. Gunja Phal Savarnam, Padmalakta Chandibam, Indragop Sankasham. So these are the certain characteristics which indicate the purity of that Artha. So basically, which is Agneya in nature. Now, wherever there is Dosh Utkatatva at that particular Matruj Bhava level also, so they may influence or interact or manifest any particular abnormal characteristics at 3 different levels. First is Bija, right? That is gametic level. Next is Bija Bhag, that is chromosomal level, and Bija Bhag Avayav means genetic level also. So any particular thing which is trying to indulge into that manifesting its own dushta on that particular Artha level will necessarily affect the Matruj Bhava. So all these Matruj Bhava, these soft organs are developed from the Artha. So when we talk about Shukra Shonit Prakriti, we need to understand there must be essentially purity of this Artha, or purity or normal functioning of all these organs, normal development of all these organs. And in the opposite side, whenever there is Dushit Artha, we may find anomalies in this particular Matruj Bhava, right? So this is the concept of Shukra Shonit Prakriti of Matruj Bhava. Again, Shukra Shonit Prakriti from Pitruj Bhava. As you all know, Shukra, the Pitruj element, paternal side, basically Saumya in nature, right? So it is also having some characteristics which indicates which is fruitful in nature. Phalavat Shukra. जैसे होना चाहिए, so bahal, madhura, snigdha, avistra, guru, pichila, shukla, bahu. So these are some characteristics which indicate the purity of that particular Shukra, paternal element, right? Gametic element, which should be Saumya in nature. So if it happens, then only the Pitruj Bhava, which are listed here, will be developed properly, will be functioned properly from the birth itself, without any congenital anomaly, right? But on the opposite side, wherever there is Dushit Shukra, there may be anomaly in this Pitruj Bhava also. So this is nothing but the Shukra Shonit Prakriti from Matruj and Pitruj Bhava point of view, right? Now, moving forward, Shukra Shonit Prakriti, if there is any abnormality at this gametic level, these are straight away responsible for development of genetic diseases, so called as hereditary diseases. So in Ayurveda, it is listed under the term Sahaj or Kulajbhav is there, Kulaj is there, Prakriti Prabhav Vyadhi are there, Adibal Pravrutti Vyadhi are there. You can see here quotation, and it is clearly indicated that At the time of conception, wherever there is dosha prakop, so we need to understand in terms of dosha utkatatva, right? So wherever there is dosha dominance, this dosha is sharir anusarpata, means we are talking about the circulation or scattered of these doshas or properties throughout the body, which will necessarily affect the garbhakar bhavas, and later on it will try to deform that particular avayav. Means organogenesis process will be hampered here by dosha prakupita, right? So Shukra Shoni, when these gametes are affected by this dosha, are responsible directly for the formation of Adibal Prukta Vyadi, which are nothing but the genetic diseases in Ayurveda. So examples are also listed there. For example, Kushta, Arsha, Prameha. So these are certain disorders which are listed under the category of Adibal Prukta Vyadi, right? Next is Kala Garbhashay Prakriti. Now this is something that is called as epigenetics, right? So where the concept of epigenetics is mentioned in Ayurveda, this is nothing but Kala Garbhashay. So when we talk about Kala, is this word is actually comprised of two different things, Kala and Garbhashay. So Kal means time. So time in terms of fertile window, time in terms of ovulation time, time for proper conception period, time for proper gestation period, right? With respect to seasonal variation. So in which rutu we are we are considering, in which rutu we are delivering, right? In which rutu, across which rutu we are trying to face our gestation period. So they are having direct impact on that particular maternal side. So this needs to be understood under the Kala Garbhashay Prakriti. Again, time with respect to the age of parent, we are having concept of Samatvagat period. Time in sense of a broader view, we are also having a special chapter in Charak Sanhita that is called as Atulya Gotriya, right? So marriages in same family disciplines are responsible for autosomal recessive disorders. We are having evidence-based papers on there. I will show in the next slide. So there are certain papers from Western sides which are exploring the effect of family disposition, right? So we will see in the next slide. Next is Garbhashay. Now this is called as Kshetra, right? Kshetra means land where the fetus is to be grown up. So we are talking about the land, and we need to understand that endometrial bed should be very well functionally active as well as anatomically well developed. So we are talking in terms of thickness, functional ability of Garbhashaya, right? So that is nothing but the endometrial bed. Along with this, Garbhashaya, what Garbhashaya is facing throughout the gestational period, how it is being developed, how it is being nourished, right? So we are talking about the practicing the Garbhini Charya. So this is nothing but the daily routine regimen which is said to be followed by a particular mother who is about to deliver, right? And in terms of abnormality, we may understand that excess exposure to radiation may hamper this endometrial bed and may lead to something, some abnormal situation. So this we need to understand from the Kala Garbhashay Prakriti, right? So this is what I was explaining in the earlier side. We are having certain modern papers also which are explaining the concept of epigenetics, and this concept of epigenetics is nothing but marking the presence of Kala in terms of season. So this is a paper having a title Season of Conception in Rural Gambia, affects DNA methylation process, right? So we are talking about the environmental impact at genetic level also, which is affecting. This is nothing but the concept of epigenetics where it is explained. So that is Kala Garbhashay Prakriti in Ayurveda, right? Again, now next is Mathurahar Vihar Prakriti. So Mathurahar Vihar Prakriti, we are very well known that whatever we are consuming, it is nothing but the Panchaputik Ahar, Shadri Sutta Ahar, Chaturvid Ahar, whatever. So that type of ahar, any particular malfunctioning, any particular adulteration, contamination of that ahar and consuming such type of ahar may develop any particular abnormality at gametic level also. So we are having Shadras Yukta ahar, but as we all know, for example, Katuras, Atiyo, excess amount of consumption of Katuyo may straight away hamper your gametes, that is Shukra will be there. So such type of examples, through these examples we can understand what is the importance of ahar and vihar at the time of gestation of that particular mother, and it directly impacts the remaining three garbhakar bhavas. The first is satmej bhava, which is nothing but the habituation or a customizational factors, which are being later on developed, are having direct impact by the help of routine activities which is followed by that particular mother during the gestation. We are having evidence-based paper also, right? We will be seeing in the next slide. Satvaj bhava, psychological factors, right? Rasaj bhava, nutritional factors. So again, whatever ahar and vihar is being followed and consumed or practiced by that particular mother during the gestational period is also having a significant role for development of prakriti of any particular individual, right? So this is what I was talking about. This is an endocrinology, journal of endocrinology, which indicates the in utero fetal programming, right? So that indicates the adverse events during intrauterine life may program the organ growth and favors diseases later in life. This is what they are talking about, the lifestyle of that particular mother during the gestational. So this is nothing but the vihar, and that accounts to the concept of maturahar vihar prakriti, right? So these are list of diseases. They have been highlighted. Vascular diseases there, hypertension, metabolic syndrome, and so on. So what they need to explore is they need to tell that there is a significant impact of lifestyle, that is bihar, what the mother follows during the gestational period, right? So this is nothing but again the concept of epigenetics only. Some more for in utero programming is there. Many human fetuses have to adapt to a limited supply of nutrients. Mathur Ahar Prakriti is shown here, which is responsible for permanent change structurally, means morphologically at the metabolic level also. So this programmed changes may be the origin of number of diseases in the later life, including coronary heart disease, related disorders, stroke, diabetes, and hypertension. So this is nothing but the significance of Mathur Ahar as well as Vihara Prakriti. So we need to understand the role of Mathur Ahar Prakriti in the formation of Prakriti of an individual, right? Last is Mahabhut Vikar Prakriti. Again, Panchabhutik Ahar, right? So whatever we are consuming, we are consuming the Panchabhutik Ahar. So Panchabhutik Ahar, all the Panchamahabhutas are having a typical ras in them, which are also having typical functional role allotted to them, right? Which are having a role in development of senses are there, and lastly, which are having a role in development of organogenesis, right? So when we talk about Akash, for example, it is having a role in organogenesis wherever there is. We are talking about the porous or hollowness of a particular organ. So that is indicated by Sarva Chidra Samuha, right? Vayu, it is indicated by Sarva Cheshta Samuha, Sarva Sharir Spandan, wherever there is pulsatile things are there, wherever activities are being taken up, wherever motor activities are being taken up, right? So there is indication of Vayu. So this depends on how the rate of activities is being influenced at the time of gestation of any particular mother, right? So this is the role of Mahabhut Vikar Prakriti. Again, the remaining Tej Mahabhut, so Santap and Bhajishnuta, right? So this again plays an important role in the organogenesis. Jala Mahabhut is there, Sarva Drava Samu is there. Optimum amount of fluidity and moisture at any particular cellular level is there. We may see many of the newborn babies are thin and cacaxic in nature, right? Which are undernourished. So when we take the gestational history, you may find that in a deficiency in the intake of liquids, right? So this shows the significance of Mahabhut Vikar Prakriti. Here accounts to Jala Mahabhut, right? Lastly, Prithvi Mahabhut. Whenever we talk about the development of an organ, we measure in terms of a fixed size, shape, dimension, right? We are talking about the Murta Swarup, and this is gained or achieved with the help of Prithvi Mahabhut only. So this is again a concept under the Mahabhut Vikar Prakriti. So what do we understand by this Mahabhut Vikar Prakriti? Whatever we are consuming, it is having a specific rasa in them, and this rasa is having specific elemental disposition in them, right? So it is made up of Panchabhutik also. So excess amount of or excess or deficient amount of any particular rasa will account to excess or deficiency of any particular Mahabhuta, and it will necessarily impact the development of any particular organ. So this is the concept of Mahabhut Vikar Prakriti. We need to understand at the time of conception how the Prakriti has been developed, right? So now types of Prakriti. Now basic classification of Prakriti is from by some different way. First, that is called as Dosha Prakriti, which is also called as Deha Prakriti or Maha Prakriti, which is nothing but the Dosha Utkatatva concept is here. That is Vatach, Pittach, Kapach is there. Again, permutation and combination, we are having Vata, Pitta, Pitta, Kapha, Vata, Kapha, and lastly Samadosha, right? Where we talk about Bhautik or Panchabhutik Prakriti, we are having 5 different Prakritis, right? Next, we talk about Manas Prakriti, which is also called as Guna Prakriti, that is Sattvic, Rajasic, Tamasic, which is again classified as 7, 6, and 3, right? Jatyadi Prakriti, this is the special concept. We need to explore something more beyond this, the words, that is Jatyadi Prakriti. This is Jati Prasakta, Kula Prasakta, Desha Anupatini, Vaya Anupatini, Kala Anupatini, and Prasatmaniyat, right? I would like to focus everybody on the suffix which has been attached here, right? Here they are talking about the Prasakta, thing which has been attached from generation to generation. So they are talking about the hereditary, right? That follows from generation to generation. They are talking about genetics is there. We need to understand Prakriti is nothing but the Ayurvedic genome. So this needs to be understood very clearly from today's point of view, right? Our contemporary science indicates that Prakriti, and which is nothing but the Ayurvedic genome. We can list that, right? And the next suffix, that is Anupatini. So they are talking about the proportionate. So they are talking about the proportionate impact of Desha, proportionate impact of Vaya, proportionate impact of Kala. Here again, Doshukkatattva concept is there. I would like to quote or bring in front of you the evidence-based quote, which clearly highlight the effect of Desh, Vaya and Kal, right? We had learned in Ashtanga a very familiar quote, that is Vayo Ahoratri Bhuktaram. So what is that? As per typical Vaya, there is Doshukkatattva. As per Ahoratra, there is Doshukkatattva. As per Bhuktanam, there is Doshukkatattva. So this is nothing but influence or impact of that particular dosha, right? But in a proportionate manner. So that comes under the Datyadi Prakriti. And last one, apart from all this, even though we understand that every identical twins are also differs from each other at some point of view. So every Atma, every Jivatma is having typical set of characteristics which keeps him odd man out. That is called as Pruthakatva. So this is called as the Pratyatmaniyat Prakriti, which is highlighted or listed under the Jatyadi Prakriti by Charaka, right? Now this Jatyadi Prakriti, we need to understand in terms of a modern term, that is Jati Prasakta. This is a racial disposition, racist, right? Familial disposition, Kula Prasakta. Desh Anupatini, geoclimatic conditions are there. Kala Anupatini, seasonal variations are there. Vaya Anupatini, age-related variations are there. Bala Anupatini, we are talking about strength, endurance of any particular related variations are there. Pratyatmaniya, individual variability is there, right? So if we try to classify this, we can go in such a way that Jati Prasakta and Kul Prasakta, we are talking about the inheritance property, right? So this accounts to internal genetic factors. Where we talk about Desha, Kala, Vaya, Bala, Anupatini, we are talking about the influential factors. Those factors, external factors, which influence in the formation of Prakriti, right? And last one, which is idiopathic. This is individual variation, that is called as Pratyatmaniya. So these comes to you, that is Jatyadi Prakriti, right? Now, the features of Vata Prakriti. I will be not going in detail in each and every feature, right? But what I would like to tell here is, when we tell students to assess, Prakriti assessment is there, we are trying to assess certain characters which try to change, which tends to change as per the situation, as per the age, as per the season. So when we talk about another concept, that is Prakriti can't be changed, we are talking, and we should understand that, and we should tell the students that we are talking, when we are talk about unchangeability of Prakriti, we are talking about genetic level. When we talk about the variations which are being changed under the impact of season, time, we are talking about the phenotype, right? So these are nothing but the triad. Whatever Prakriti assessment formats are available till date, we are assessing only the phenotypic expressions. These are triad, and this tends to change as per the situation, what we favor or what we prefer as per the situation, right? So all these characters which are listed in Vata Prakriti, Pitta Prakriti, and Kapha Prakriti may tend to change as per the situation. So this is again a concept of epigenetics. This is again a concept of Dosha Utkatatva, and this is nothing but the phenotype or triads which are being expressed in front of everybody, right? Now we will study factors which are essential for development of congenital anomalies. So when we talk about congenital anomalies, we need to understand that talking in terms of three different levels. We are talking about gametic level, that is Bija. We are talking about chromosomal level, right? That is Bij Bhag, and we are also talking about the genetic genes level, right? That is Bij Bhag Avayav. So the clear-cut reference are also mentioned here, which is in front of you on the slide. That is SHARIR chapter 3, which indicates. Now I would like to focus everybody on this word, that is Upatapto, right? So we are talking about the Dosh Utkatatva as well as Upatapto, means doshas by the virtue of their guna are trying to mark their presence at these three levels, Bija, Bij Bhag and Bij Bhag Avayav, and which are essentially trying to impact organogenesis process, right? And they affect, see here the word comes Shonit and Garbhashay. So they are affecting necessarily the maternal gamete also, they are affecting the Garbhashay, that is Kala Garbhashay we had seen earlier. So this is nothing but the concept of Shukra Shonit Prakriti and Garbhashay Prakriti, right? So we need to understand the role of dosha in formation of congenital anomalies, right? The same is highlighted in this shloka. I will be moving forward. I will be not explaining this shloka here, but the same is depicted in the table here, right? So when we talk about bija, we are talking about gametes. For example, in just an example, modern diseases, we are talking about azoospermia is there, necrospermia is there, right? So when we talk about cause for this anomaly, there is combined formation of Bij and Bij Bhag, right? So wherever there is anomaly in the Bij and Bij Bhag, Ayurveda says that in case of males, they had listed Vandya. In case of females, they are also listing Vandya. So this accounts to infertility, right? When we talk about Bij Bhag, that is nothing but chromosomes. As Dr. Pawar has already mentioned in detail about the chromosomal anomalies, I would like to highlight only 2 to 3 few names in front of this, because we are having focus more on Ayurvedic Prakriti. So chromosomal defects, Down syndrome, Turner syndrome, Klinefelter's, and the list is endless, right? So this is possible due to the anomaly at the Bij Bhag and Bij Bhag Avayav level, and this represents or is manifested in males by the Puti Praja, in females by Puti Praja. So what do you Puti Praja mean? It indicates dead fetus, or it indicates the fetal anomaly, right? So these ultimately the doshas by the virtue of guna are responsible for this only. And the last, Bij Bhag Avayav, which are nothing but the genes. We are having single gene defects are there, multifactorial, that is multiple gene defects are there, which is due to the anomaly at the level of Bij Bhag Avayav. Now here comes the word Ekadesha. They are talking about any particular component, right? As Dr. Pawar has also mentioned here about the locus, right? So the position of gene is having an impact on development of hermaphrodite nature. So the hermaphrodite, this concept is being highlighted in Ayurveda with the name that is called as Trunaputri in males, Varta in females, right? So this is nothing but the factors which are responsible for development of congenital anomaly, right? Now we will be having an outlook for Garbhakar Bhavas, Matruj Bhavas. So we need to understand Matruj Bhavas, the role of Matruj Bhava in terms of Kula or Gotra of parents, as I told you earlier, Atulya Gotriya. Maternal age is very important for time of conception. Right? Health of the reproductive development of reproductive organs is very important. So we need to understand the healthy status of reproductive organs, time of conception, beige of that mother, that is gametes, and maternal diet during the pregnancy. Now this is the gestational age. So all this accounts to proper development of Matruj Bhava. So we are talking about the organogenesis. So organogenesis accounts to the morphological development of particular Prakriti, right? So lastly, I would like to tell that about the Atulya Gotriya. So there are certain evidence-based papers in Western medicine, Western schools of medicine. They are highlighting the rates of chromosome abnormalities at different maternal ages. It means that they are talking about the age of mother, right? Maternal ages and the rate of chromosomal abnormality, right? So this is nothing but the Atulya Gotriya. They are talking about high rate of traditional consanguineous marriages in certain tribes, races, and religions, which happens usually at African races, which increase the frequency of autosomal recessive disorders. So what we talk about, Matruj Bhav organogenesis has been impacted or impaired when beyond proper age, समत्वगत वीर्य, ये कंसेप्ट छोड़ के अगर प्रॉपर एज के बियॉन्ड अगर कंसेप्शन होता है, तो दिस हैज अ डायरेक्ट इम्पैक्ट ऑन क्रोमोसोमल abnormalities, right? Pitruj Bhav, we are talking about the development of all those organs which accounts to Pitruj Bhav, the paternal side. So these are developed from the Shukra, which is sound in nature. So whenever there is uplifted bija, sperm, as well as abnormalities in Shukra and Vayu, so the progeny with congenital genetic anomalies will be seen there, right? So this is also quoted in the modern Western schools of medicine. The paper is there. It shows that the increased mutation rate is due to accumulation of new mutation from many cell divisions. So the more the cell division, more chances of error or mutation occurring there. So genetic defects accounts to vitiation of Vayu. As we all know that Vayu vibhajati. So Vayu Mahabhut is responsible for the division of cells, right? So this is nothing but the clear-cut evidence which is shown in Ayurveda also, and that is having an impact at Pitruj Bhava. Also, there is mentioned that advanced paternal age logarithmically increases the risk of new mutation, and which is a cause for autosomal dominant diseases. So this is again a concept of Samatvagat Virya, as we had seen in the previous slide also, right? The next one is Atmaja and Satmaj Bhava. So we are talking Satmaj more precisely here. We are talking about the habituation. We are talking about the customizational factors of one's own constitution, habitat, time, season, wherever he is dwelling or she is dwelling, right? So we are having Kala Satmya again, Desha Satmya is there. So this is nothing but the concept of epigenetics, which is mentioned in Ayurveda. So improper time, season, age of conception, all these periodical factors can influence the health of fetus by creating mutagenic or epigenetic influence. This is nothing but the concept of epigenetics, right? So what I would like to tell is genetics starts from Ayurveda only. Genetics where the concept of genetics is mentioned in Ayurveda, that is nothing but the Prakriti. That is nothing but the Garbhakar Bhava. That is nothing but the factors for formation of Prakriti, right? Rasaj Bhava. So Rasa, when we talk about the nutrition, we are having a concept of Masanumasic Ahar. Month-wise diet is there, right? Which is to be followed by a particular Garbhini. So gestational age, during the gestational age. Here we will take an example. For example, excess consumption of Rukshaguna is there, or rather suppression of urges is there, which leads to Vata vitiation. So this, in case of Vata Prakriti, we may see Utkatatva of Vata, Utkatatva of Rukshaguna. So this Rukshaguna is basically restricting in nature, restrictive in nature, excess absorption is there, so which will hamper the normal qualities of Shukra, right? So there will be Nishpalatva rather than Palavat Shukra. So this is called as Sheena Shukra. And this may lead to Alpa Apatya, which is a classical symptom of Vataja Prakriti, right? Again, Rukshaguna, which is restrictive in nature, which opposes the movement, free ease movement is not there. So it may lead to Pricha Vyavahita, it may lead to hamper the normal phonation or act of phonation process. So we are having Pratat Ruksha, Jarjar Swara is there, right? So this is possible only because of Guna. So this indicates the Utkatatva of Guna. Lastly, Ruksha Guna, excess absorption, this may lead to malnutrition, and we are having Rukshan Apachit Sharir as a classical feature of Vataj Prakriti. So this is the concept of development of a phenotypic triad by a Guna, right? So these are the Rasaj Bhava. Sattvaj Bhava. So human, I believe that human being possess a basic instinct and intelligence, right? So manas or psyche is nothing but the instrumental form just to experience all those things. So sattva of any particular fetus can be studied or can be developed by these three things. First, by the sattva of parent, which is nothing but the genetic inheritance or genetic derivative. Garbhini Charya, what that mother suffers or faces during the gestational derivatives, and Desh, Kala, etc. from the conception till delivery. Conception through gestation till delivery. So these are the epigenetic derivatives. So all these comprises the psychological understanding of every particular cell, and lastly the garbha, right? So this accounts to the formation of Prakriti. We are having, as Dr. Pawarkar had already mentioned in detail about the gene, chromosome, and gametes, I will be having just an Ayurvedic point of view from genetic level. So here, as we all know, heredity is the process of passing genes from generation to generation, right? So chromosomes, which are the basic unit of heredity, where the genes are arranged in a linear form on over the DNA, right? And genes are only the segments of DNA which are responsible for controlling the diet, right? So genes, nothing but which comes in different form, and that forms are called as allele. Allele is nothing but a different version for a gene. So we are having dominant allele, we are having recessive allele, which are responsible for expression. Now, from Ayurvedic point of view, when we talk about dominance and recessiveness, we are having genes which is nothing but the Bij Bhag Avayav, which is having dosha Utkatatva, which is having dominance of dosha, dominance of guna, and as you all know, guna are nothing but the conglomeration of Panchabhutik elements. So in Panchabhutik elements, we are already learned about these three concepts. Paraspar Sansargata, when we talk about any two particular matter molecules which are coming together, the particle nature will be, right, any of these three. Paraspar Sansargata, they may be contagious. Paraspar Anugrahata, they may follow each other, and Paraspar Anupraveshacha, right? So any of the thing is dominance. So when we talk about this dominance, we are talking at the genetic level about alleles, and we are talking about the expression of that particular triad, right? So this is the Ayurvedic lookout towards the gene. I would like to try to explain this with an example again. Consider if you are trying to explain a triad or a disorder which is caused by a gene on Y chromosome, right? So it will appear in males of the family, and it will pass from father to son only, all the males from generation to generation. So daughters will neither inherit nor pass on the disorder. If we consider a triad or disorder which is caused by a gene on X chromosome, right? So again, we will find that it is much more common in males only. Why? Because females have two X chromosomes, and therefore two copies of gene, one of which would mask the influence of another. So, and this is nothing but the development of X-linked triads are there, right? So this is a congenital anomaly. But we need to talk about masking of influence, right? So when we talk about influencing each other, we are talking about the dominance, we are talking about following nature, we are talking about the contagious nature of any particular two properties which come together and trying to impact the cell, right? So this is nothing but the genetic genes. Genotype, all the organisms are having a basic genetic material which is called the genome, right? So individuals in present throughout the world are having a different combination of alleles. So genotype is nothing but the combination of alleles which a particular individual has. So the genetic makeup of a triad is nothing but a genes, which are again present in three forms. Homozygous dominant is there, homozygous recessive is there, and heterozygous. We need to understand when we talk about samadosha prakriti, we are talking about the homozygous nature, not heterozygous. And when we talk about heterozygous, this is the dosha utpatatva, right? So this is the dominance of any particular dosha, dominance of any particular guna. And when we talk about this allele, these are certain things which forms the genotypic or genetic constitutional makeup of a particular individual, which remains consistently throughout the lifespan, and which is again quoted in Ayurveda also, prakopo va anyatha bhavo kshayo va na upajayate prakrutinam swabhavena jayate tu gatayusha. Means what we talk about unchangeability of prakriti is nothing but a genotype. And in Zatyadi prakriti, we are having two things, that is Jati prasakta and Kula prasakta, right? Phenotype, these are nothing but the expression of genotype, the way we look or appear. So, for example, height, for example, hair color, eyes color, right? So, it is been expressed only because of the dominance. So, when we study about dominance, we are having two things. First is complete dominance. When a dominant allele always gets its way, right? Means that even though no matter whoever is its partner, but the dominant allele, if it gets its way, it will show its complete dominance, right? And incomplete dominance, dominant triad don't completely get their way. It means they are trying to compromise themselves. So here we are having mixed phenotype is there. Another word is co-dominance, right? That also accounts to expression of a particular phenotype. So this is nothing but the expression of a triad that is called as phenotype. Allele, as I had told you earlier, a version of a gene. There is a gene that controls eye, for example. So we are having separate alleles for each other, different colors of eye. One allele for blue color, one allele for brown colors are there, and this comes from gamete. So every haploid cell carries one allele, right? So diploid cells, when we talk about diploid cells, they are having two alleles, and this is necessary that there must be a pair of genes for expression of triad. It means we need two alleles to express, right? So this is what we are talking about Shukra Prakriti. This is what we are talking about the dominance. We are talking about the Dosh Utpatakva, right? Another new word in today's era, it has been very familiar with most of the Ayurvedic physicians, and I would like to tell all the students that get familiar with this word, that is Ayurgenomics. So Western science of medicine, Western schools of medicine are trying to have a different approach to look towards Ayurveda, right? So they are trying to find the base of genetics in Ayurveda. So study of genomes is nothing but genomics, and genome is the genetic material present in an organism. So they are trying to coin a new term that is called as Ayurgenomics, which means application of genomics to the basics of Ayurveda, mainly to the concept of Prakriti. So if anybody asks you where the concept of genetics is explained in Ayurveda, the first word which should come in mind, that is Prakriti. So this is what the new term has been arised, which is a hope for diagnosis and treating a disease, right? So I am keeping a open question for all the viewers here, that Prakriti should be considered Ayurvedic genome or not, right? Another word they are using nowadays, that is P4 medicine, which Ayurveda used to prefer from since ancient times, right? What P4 indicates? Predictive. So Prakriti is, when we know Prakriti of any particular individual, we may try to predict any particular disease, we may try to predict any particular development of a triad also, right? Preventive, when we know Prakriti of an individual, we try to prevent the future developed disorders. Personalized medicine approach, which is again Prakriti. And lastly, most important word, that is when we try to assess Prakriti of an individual, we are trying to indulge that individual to actively participate to know himself, right? So active participation of that is a new approach towards medicinal fraternity. So this is participatory medicine. So all these 4P accounts to P4 medicine, which Ayurveda used to practice since ancient times. Another word, that is nutrigenomics. So Western schools of medicine also having an impact, also having an outlook that nutrition is another factor, important factor, which affects the genetic constitutional makeup of an individual. So they had coined a new term, nutrigenomics, right? And for that purpose, it is they are coining another term, that is physiome. Physiome means the set of functional characteristics which is only dependent on the type of nutrition whatever we are consuming, right? So this Ayurveda inspired concept of personalized nutrition is a novel concept of nutrigenomic research for developing personalized functional food, nutraceuticals suitable for one's genetic makeup with the help of Ayurveda. This is the particular paper published in Journal of Traditional and Complementary Medicine. I had quoted here, right? So this is another new concept of nutrigenomics there. Epigenetics, there are certain papers which highly are evidence based, which show the role of time, age, season, right, which comes under the concept of epigenetics. So maternal and paternal chromosomes are not only responsible for phenotype, but epigenetic factors are involved. So we need to understand that environmental cues trigger changes to epigenetic taxon genome, which shape the way genes are being expressed. So we are not inheriting the genes, but potentially we are also inheriting the modes of expression, whether it is to be expressed in this generation or in next generation. So this depends on the favorable environment, favorable age, favorable season, all these things. So this is nothing but the concept of epigenetics in Ayurveda, right? So again, where we talk about epigenetics, there is an evidence-based paper which shows that DNA methylation is what occurs when methyl groups and epigenetic factor, which is found in dietary sources, means we are talking about Mathur Ahara Prakriti, right, can tag the DNA and activate or repress the gene. So expression or suppression of genes is dependent on Mathur Ahara Prakriti. So this is the nutritional approach towards the Prakriti, right? Lastly, I would like to explore the significance of Prakriti. So this is what the Prakriti should be understood. The importance of Prakriti should be understood. Assessment of healthy status of an individual can be done by Prakriti, evaluation of Nadi Pariksha, evaluation of Agni and Kosha to predict the prognosis of a disease, to predict the course of a disease, susceptibility or resistivity towards the disease. It is important from diagnostic point of view. Again, Prakriti is important from choosing the Rasayan therapy also. Prakriti can be used as a tool for therapeutic point of view also. Most importantly, which is used nowadays, that Prakriti can be used as a key parameter for development of a strong social relationship. Social behavioral therapy is there. It is a tool for choosing the profession also, right? So it is actually a marker to understand the appropriate choice of life partner is also there, right, for marriage. And it is also a tool for making necessary arrangement for designer babies also. So signature babies or designer babies is another concept which is been highlighted in today's era, and which is completely dependent on the nutritional gestational age, Mathura Harvya Prakriti, Kala Garbhashay Prakriti, right? So when we talk about a single term Prakriti, what I found is the list is endless, but I had highlighted here some of them. The Prakriti can be there are number of researches which are ongoing and which are completed. Prakriti and Ayurvedic genome, Prakriti and single nucleotide polymorphism is there. Prakriti is related with the rate of development of autosomal recessive disorders is there. Prakriti and epigenetics, Prakriti and Ayurgenomics, Prakriti and autonomic nerve responses, Prakriti and Parkinson's disease, irritable bowel syndrome, and number of diseases are there. Prakriti genome-wide analysis is there. And here I would like to mention specifically that Dr. Sarita Budada is with me, that is a professor at this college where I am working right now. And I and Madam had consistently worked since last 6 years on different small-scale experimental researches on Prakriti and Lalat, Prakriti and Bahu, Prakriti and Vaksha, Prakriti and Pindika, Prakriti and Varna, Prakriti and Netra, right? So Prakriti and Dhatu Sartha is there, Prakriti and intelligent and emotional quotient is there. So this is how we can understand the significance of Prakriti, right? Lastly, I would like to keep comparative things on at a same platform. William Sheldon, if you had known with this person, William Sheldon in 1940s, right? In 1970, he has wrote in his book, that is Atlas of Men, where he tried to categorize all the people in a single somatotypes, right? So what he found, he has done measurement on terms of muscle with the help of size, strength, and endurance. He has measured the bone in terms of length and girth. He has measured the fat in terms of percentage and distribution. So the ultimate findings of all these can be stabilized in a single manner. We are having germinal layers with us. We are having three typical body type with us. Endoderm is there, endomorph is there. So endomorph represents the phenotypic presentations which are responsible for digestive tract activities. So such type of individuals are called as viscerotonic. And when we talk about organogenesis, so endoderm is again responsible for development of gut and visceral organs. So this is nothing but that when we talk about gut and visceral organs, we are talking about nutrition, we are talking about portion, we are talking about color, right? So this is nothing but a kapha. So this accounts to Kapadosh Prakriti. And another scientist is there that is called as Krishmer's. So Krishmer's classification of personality ke hisab se, it is been classified under the type of picnic type of personality. We talk about mesoderm, we talk about mesomorph, where it indicates the structural buildup of muscle, functional activity of circulatory system. So such type of individuals are called as somatotonic, right? And mesoderm is responsible for development of again muscles and connective. So see, this is a direct connecting link of body type and germinal layer, right? So muscles and connective tissues there, which accounts to metabolic activities. So this is nothing but the Pitta Prakriti, and according to Krishmer's classification, athletic type of personality are accounted under the Pitta Prakriti. Again, last is ectoderm. Here, ectomorph, nervous system phenotypic expressions, understanding capability, grasping power is there. So we are having certain features, right? Under Prakriti. For example, we are having Shigra Samarambha Kshobdikaraha, we are having Shrut Grahino, quick grasping power. So that accounts to what Prakriti. So this indicates the clear-cut connecting link. So such type of individuals are called as cerebrotonic. And or in terms of genetic organogenesis, we are having epithelial tissue, nervous tissue, and nervous system from ectoderm. This is nothing but Vata Prakriti, and according to Krishmer's classification, these are lean and thinner persons. These are called as asthenic type of Prakriti. So on a single platform, we can categorize somatotype, genotype, phenotype, as well as dosha Prakriti, right? So scope of research in Prakriti, I will be just giving a scope in front of you, and I will finish it off. The Prakriti in Ayurveda relates with organogenesis, it relates with histological dominance, it relates with epigenetics, genomics, ayurgenomics, nutrigenomics, genotyping, phenotyping, genetic engineering, P4 medicine, immunity, designer baby, right? So all these are nothing but a scope, wide scope of research in Prakriti in Ayurveda. So these are the references which I had gone through for today's lecture. And with this, I would like to thank Dr. Sandeep Bilari, who had recommended me for today's session, Dr. Pramod Kanab sir, Dr. Syed Mushtaq sir, Dr. Dipali Patil madam, and all the organizers and teaching fraternity from L.R.P. Ayurvedic Medical College and Hospital from Islampur, Sangli, Maharashtra. I would like to also thank Dr. Sarita Bhutala madam and GGPSR, my own institute, and CVM University for providing me such an environment to present in front of you all. So with this, I end up with my session, and thank you. Thank you once again.
For your very informative lecture. Session was very nice with regards to yes handling of the concepts like with respect to genomics. Again, the Prakriti related some terms has been very Matu Ahar Vihar Prakriti, Prasakta, and Anupatini were the nicely elaborated terms from the sir. Shukra Shonit Prakriti, Kala Garbhashaya Prakriti, where has been handled by the sir as epigenetics. Punnett square, harmless nature of the Prakriti were also being expressed very nicely and delivered very perfectly by Chilwan sir. I am very much thankful to the sir for this elaborative and very nicely presented lecture.
Now, I would like to invite Dr. Aruna Bale ma'am, Principal, Professor and HOD, Department of Kriya Sharir, Gomantak Ayurved Mahavidyalaya and Research Center, Shiroda, Goa, on this platform for expert opinion and summarize both the sessions. Please continue, ma'am.
Good afternoon. Good afternoon, everybody. Respected dignitaries, Dean, Principal, Vice Principal, Director of Post-Graduation, Dr. Khanna. Let me first take the opportunity to express my gratitude for giving me the opportunity to chair the session. To chair both the sessions, in fact. And since this is organized on the occasion of the birthday of the founder member, Nishant Patil, I express my birthday wishes to him. Coming to the point of the first speaker, Dr. Sudhir Pawar, it was really informative on genetics and role in medical education, wherein he has expressed genetics in the era of present and future. He has also said very elaboratively on the biochemical properties and different branches of genetics. Also important role on hereditary. Hereditary, how it is controlled by genes. On this, I would like to little bit simplify according to Ayurveda is, according to our Indian philosophy, the Srishti Nirman Parampara, evolution of the universe, was understood on the basis of Karyakaran Siddhanta. That is the theory of predicting the Karya effect by looking at the cause, that is Karan. Anuvanshik Siddhanta is one amongst the Karyakaran Siddhanta which is based on predicting the Karya by understanding the Karan, that is, future product will be Anurup of the Karan, similar to the cause. The concept of this Siddhanta is very similar to the concept of inheritance. This Siddhanta propounds about the factors such as Bija, Bija Bhaga, and Bija Bhaga Avyava, which influence the occurrence of hereditary disease, deformities such as the blood disorders like thalassemia, hemophilia, sterility, other sexual perversions. So, sir has given very well elaborate, informative description on the genetics. I congratulate the speaker for this important session, important deliberation given by Dr. Sudhir Pawar, sir. The next speaker, Dr. Aniket Shilvant, who has very well elaborated on the Ayurveda and genetics, wherein he has given the relation of Prakriti, how it can be related with genetics. He said, Doshanu Shaita Esham Deha Prakriti Uchyate, which is a very good important reference given, Anushaita Esham, that the factors responsible, how the Prakriti is formed on the Utkatatva of gunas, qualities, how Prakriti is related and dependent on the qualities, Utkat qualities of particular dosha. Also, he has given Paraspara Anugraha, Paraspar Samsarga, Paraspar Anugraha, Parasparanu Pravesha, which is also very important with relation to genetics. And also epigenetics, he has explained, and different types also, how Prakriti can be related, Dosha Garbhotpakara Bhava, like Rasaja, Satmyaja, Sattvaja, how they are formed by considering how we can consider the different Utkat qualities in this, and how not only the Shukra Shonit is responsible for the formation of the Prakriti, but also dependent, the formation of the Prakriti is also dependent on the Kala, dietic regimens, behavior of the mother, nature of the Garbhashay, and on Mahabhutas. This is very well explained by Dr. Aniket Shilvan, sir. So I congratulate the speaker. Now I'm putting forth my views on this particular genetics, how we can correlate it with Ayurveda. See, what I feel is when we say Bija, Bija Bhaga, Vyava, and here we say it is genetics, Bija, that is gametes, Bija Bhaga, chromosomes, Bija Bhaga, Vyava, we relate to genes. If the moderns, they are continuously on the research, if the further research is made on the genetics and they come to a smaller fraction on this, how you will relate it to Bija or Bija Bhaga or Bija Bhaga Vyava? Because Ayurveda has considered Bija Bhaga Vyava only. So should we come to the Parmanu level? Should we relate it to the Anu level? This is one point. And coming back to the main, when we say it, Ayurveda has very well explained Jati, in Charaka also has explained Jati Sutriya Adhyaya in Sharir Sthana. See the word gene, looking at the word gene, the Sanskrit word Jana, Janma, Jati, wherein you will find astonishing similarity between the pronunciation and their meaning. Well, all of us, we know that genetics, it's a branch of reproductive science and deals with the similarities or deals with the studies of the factors that are responsible for the transmission of similarities and dissimilarities from parents to the offspring, and phenomena of transmission is responsible for the continuation of race and species known as jati in Sanskrit, wherein we can find very good reference about this in Kashyapa. Kashyapa has said, Purusha Purusham Abhinirvartayanti, Gorgama Ashva Ashvam Evam Adi, meaning that the human being reproduce human beings only, and the same is true for bulls or the horses or the other animals also. Charaka has also written in his Jati Sutriya Adhyaya about the same. The Chakrapani, the most brilliant ancient scholar, the commentator of Charaka, also explains about this, wherein when he gives the commentary on Charak Sharir Stan 2 and Charak Sutra Stan 30 also, Jati iti deha jati, meaning the word jati is related to the body and its figures and the phenomena of transmission of bodily configuration and new creation occurs to the same secret code line. We have one more reference given by Chakrapani. Manushya bijam hi pratyanga bijabhaga samudayatmakam swasadrusha pratyanga samudaya rupa purusha janakam. So here also we can say that this means that human bij, the germ cell or the sexual gametes assemble in it in a manner of fragments representing the different tissues or the organs of the human body. And these fragments particular are responsible for the transmission of the similarities from parents to offsprings. When we come to the another commentary by Sushruta, the Sushrut has also explained this, that Shukra Shonita Garbhasayasta Atma Prakruti Vikara Samurchitam Garbha iti Uchyate in its Sutra Sharir 4, meaning that the Garbha is applicable, the term Garbha is applicable only to a mass in the uterus that is formed as a result of perfect union of male and female gametes and further joined by the Atma, the life force. Here we can scientifically use the definition of Garbha. It's the perfect union, as it is also said in Prakruti definition, Shukra Shonita Sanyoge Yo Bhaveto Shukrata Prakruti Jayate Tena. So here it's a perfect union of male and female gametes, which include that everything is present inside them. So what is the reason then behind the emphasis of Prakriti and Vikaraha? So the reason is that the Prakriti, especially the Vikar and the perfect interlocking is a very important step in the formation of Garbha, because these are the factors which maintain the unity and the in diversity and diversity in unity among the species and the individuals. The unity in diversity means that even though the inherited characters that accompany the male and female gametes differ drastically, the same features of the species are still maintained in each and every species. This phenomena is known as Jati Prasakta in Sanskrit. Diversity in unity means that the children born from same parent may show marked variation in their skin color, height, strength, and intelligence. And this phenomena is called as Pratyatmaniyata, that is individual identity. As it was said by Dr. Shilvanta, Pratyatmaniyata. So here when we say Jati Sutra, can be said that it is imprinted code language within the sex gametes, while the Prakriti and Vikara are imprinted code words in the Jati Sutras. The reference we can quote from Sushruta is Prakrutya Pradhanadaya Ashto Vikaraha Panchabhutani Ekadasha Indriyani iti Shodasha. Meaning Prakriti are 8 and their precursors are subatomic, biophysical and biochemical states of Panchamahabhutas. And the Vikaras, we know they are 16. So that is 5 structural constituents of the human body: Pancha Jnanendriya, Pancha Karmendriya, Mana, that is the sight center, that is the main instinct. Thus, the Prakriti and Vikruti include everything which will help us to identify the species and the individuals. For this, even Dalhan, the commentator of Sushruta, has given a very good reference. Jati prasakta nama tasu tasu jatishu te te karacharanadaya swabhava visheshaha vachaneshu na cha nanavida evam vyakta vyaktadayo. For example, that is karacharanadaya, that is hands and feet are the extremities, they differ species to species. Swabhava vishesha, inborn instinct, that is related to mind, they also differ from species to species. Vachaneshu vividata, that is diversity, speech diversity also differs. Ahar, that is eating habits, likes and dislikes, they also differ from species to species. And pratyatma niyata, finally, that is individual diversity. That also children born from the same parent differ drastically in their skin color, height, strength, etc. and intelligence. Now, when I said about the Parmanus, also there are references I will give you. Anu pravana bhavacha shukra dehat pratichyate charak chikitsastam. That is shape, size, constituents and probable number of Jati Sutras and the sex gametes, meaning that the important work attributed to Shukra Dhatus is to produce bija. This, the bija is Anu or the microscopic in size. Even Kashyapa has said, Pranaktu bija dhatu hi vibhajati ati sankhyayaha pratishta matram tu bijam hi rajatena pariveshtite. Meaning that the motile male gametes are produced in huge number by the process of division, but only one enters the female gamete, which in turn encircle it and form from all the sides. Thus, the bija is microscopic, and every time this male bija produced in huge number, and bija is anu inside, that is jati sutranu. This anti-shukranu, that is also known as param shukra anu or parmanu in Sanskrit. That is parmanu nam sayoga kurvan shariram arambhate, Charak Sharir San 7. That parmanu union is the keynote for the construction of the human body. Another reference you will find in Charaka is Atindriyaste ati sukshma rupa atma kadachit na viyukta rupa, meaning atma, the life force, unites with the human body at this anti-sukshma level and the life process begins. And this concept is very true even today. Another, you can say, the next question is, then how these Mahabhutas are arranged within this Jati Sutra Parmanu. Here also another reference given by Charaka is, Syat Chatushpada Prabhavam Vayur Agni Bhumi Ambu Guna Padavat Chatushpadadi Prabhavam Sukshma Bijasya Janati, meaning the arrangement of these four Mahabhutas is like four legs or limbs, and that too according to their most natural but dominant gunas. Now, when we say this, we can see that the most natural but dominant gunas out of these four Mahabhutas are to clarify the position and direction of these limbs. Here, the Sushruta has given a very good reference. Prithvi Ambu Guna Bhuyisthani Gurutvat Adho Agni Vayu Guna Bhuyisthani Lagutvat Urdhvam. Meaning, Prithvi and Aap always denote downward direction because they have Guru, that while Teja and Vayu always denote upward direction because they are light, Lagu. So this, as described, that is the example here, participating in the construction of Jati Sutra Parmanu. Here another, even we can say, Akash being inactive, do not form a limb, but it has remained in an inactive position because it is Rupa Rahita. Teja and Vayu form the upper limb, whereas Prithvi and Apa forms the lower limb. So here this will Chaturbhuja Madhya Kila Madhya Vimukta Agro Swastikam. That is also one more reference from Bhagavata Sutra. That is the shape of Jati Sutra Parmanu is like swastik, or in short, Jati Sutra Swastika Parmanu. So regarding the number of Jati Sutra in each and every sex gamete in the human being, there are two schools of thoughts. That is Charaka. According to them, they are 56, and according to Sushruta, they are 23. Manushya Bijam hi Pratyanga Bija Bhaga Samudayatmakam, that is said by Chakrapani in Charak Sharistan 3 commentary. That is, the human Bija sex gamete is a cluster of Bija Bhaga, and this Bija Bhaga represents the tissues and organs of the human body called Pratyanga in Sanskrit. Therefore, one Bija Bhaga must represent either one Pratyanga or one Pratyanga Bija Bhaga Vibhaga. So thus the number of pratyangas, that is pratyanga vibhagas, will coincide with the number of bija bhaga, that is the Jati Sutra basis of the counting. So here what I can conclude is that Jati Sutras are the Parmanu, which are smaller, subtle in shape, size, but we can say, as per the reference given in Bhagavata also, that it is swastik in shape, Panchabhautik in structure, and probably 23 in number according to Sushruta. Even 23 they have counted in moderns. So this is what I would like to conclude about the genetics given. Most of the time, genetics, naturally we say Prakruti, it is formed Shukra Shonita Sayoga, and that depends on the Guna Utkatata, which we correlate with the genes. Another, as said by Charaka in the Charika Sharistantri, we say thus, that is Bija Bhaga, Bija Bhaga Vyavasya, that is we go subtle. Here also, that was very well explained by Shirvanta, considering the Yoni Vyapat also, how it can be said Putija Yoni, even when it is said there is deformity in the male gamete and male Bija Bhag and Bija Bhaga Avyava, the Trina Putrika Yoni is considered. So this way, there is very description given, but here we can correlate this, according to me, we can correlate this with Jati Sutra, how the Jati Sutra Adhyaya explained by Charaka and Jati Sutrani, that is difference in the Parmanus. Thank you. Thank you once again for giving me the opportunity to express my views.
Thank you so much, ma'am. Thank you so much, madam, for your valuable guidance and conclusive opinion. Link for feedback is shared in Zoom chat box, also in YouTube chat box. Now I would like to invite Dr. Pramod Kanap sir, PG Director and Associate Professor, Department of Kriya Sharir, for vote of thanks.
Good afternoon, everyone. On the auspicious occasion of birthday, Honorable Nishikant Dada Bhosle Patil, founder of our Prakash Shikshan Mandal, Department of Kriya Sharir, Lokneti Rajan Bapu Patil Ayurvedic Medical College Hospital, PG Institute and Research Center Islampur, has arranged the national webinar on genetics and Ayurveda. On behalf of our institute and Department of Kriya Sharir, a big thank you to today's eminent speaker, Dr. Sudhir Pawar sir, Professor and HOD, Department of Anatomy in Dr. Vittalrao Vikhe Patil Foundation Medical College, Ahmednagar. Dr. Aniket Shilwan sir, Assistant Professor, Department of Kriya Sharir, G.J. Patel Institute of Ayurvedic Studies and Research, Anand, Gujarat. Dr. Anura Bale Madam, Principal Professor and HOD, Department of Kriya Sharir, Gomantak Ayurvedic Mahavidyalaya and Research Center, Shiroda, Goa. I would like to appreciate all of you for offering your service and time to deliver lecture in the webinar. The institute and our department appreciate your work and seek the same from you in future. Thank you, sir. I also extend my thanks to Sri Nishikant Bhosle Patil Dada, founder of our Prakash Shikshan Mandal Islampur, for their valuable support and guidance. We are thankful to our chairman Sri Sanjay Jadhav sir, secretary Sri Nitin Patil sir, and Sri Sandeep Yadav sir for their support. Our department is very much thankful of respected Dean Sir Dr. Virendra Minkire sir for their timely guidance and cooperation in organizing the webinar. Our department is greatly thankful of Vice Principal Dr. Ranjit Mohite sir, PhD Director Dr. Hausarao Patil sir, Hospital in charge Dr. Vishal Patil sir for their guidance and help. We are deeply grateful and thankful of all the respected faculties of our institute and other institute for their active participation in the webinar. Thank you very much all PG and UG students for your active presence in the webinar. We are fortunate enough to be backed by the team of motivated and dedicated ICT department, Dr. Ajit Patil sir, Sri Abhijit Patil sir for their hard and valuable work. Thank you very much, sir. Thank you one and all.